Welcome everyone to session number 5 of the CERSI course. Today we have a lecture on clinical biomarkers and companion diagnostics. Next slide, Nathan. And this flows naturally from our previous topics. And today we're going to focus on explaining the different types and applications of biomarkers in clinical drug development. Next slide, please. Again, another way of looking at the session flow. We have 3 wonderful speakers today: Heleen Scheerens, Priti Hedge, and Rhea Nersesian. And if you look on your screen, you can see that they're all together in one room. I think this is just so incredibly special. And in fact, Priti Hedge is actually visiting Genentech to make this possible. So we really, really appreciate that. The next slide. Session 4, Monday, session 5 today. The recordings will be posted next week. We had 2 sessions this week. That is a lot. And we appreciate your commitment to the course by joining both sessions. There is no class tomorrow, Friday, so that's sort of the upside of having 2 sessions this week. No session tomorrow, Friday. Again, as always, the Q&A in Slido, and I clarified with the presenters today, they want to keep this fluid and interactive, so they're willing to take questions on a regular basis, and there's 3 of them in the session, 3 instructors, so they can support each other with the Q&A. So feel free to use the Slido to ask questions. If you want, you can also raise your hand. I imagine that would be fine as well. Try to keep your camera on. That always helps with engagement. And then one thing that Nathan reminded me to mention is that in the session that we have next week, there's going to be something slightly different. So we don't want this to surprise anyone, although I think it's a good surprise. The instructors have decided that for the last hour, they're going to have 3 breakout rooms and offer 3 different flavors of lectures. Is like a choose your journey situation. So that's coming next week. Next slide. I'll start with introducing the speakers today. Our first speaker instructor is going to be Dr. Heleen Scheerens. Heleen is the Vice President of Global Head of OMNI-Biomarker Development at Genentech. She has 20 years of experience in pharmacology. In her role at Genentech, she leads a diverse group of more than 60 science professionals, and she manages and develops biomarker strategies for this therapeutic space, which is abbreviated as OMNI at Genentech, and it includes the therapeutic areas of ophthalmology, metabolism, neuroscience, immunology, and infectious diseases. So I never realized that it's a double E, so OMNI. That's great. She's also actively involved in GY's, Genentech Women in Science and Engineering. Before joining Genentech, she held leadership positions at Celera and Rigel Pharmaceuticals, and she has a PhD in immunopharmacology from the University of Utrecht in the Netherlands. Next slide. Our next speaker, and she is a new speaker to the course. Thank you for joining us, Priti Hedge. Dr. Priti Hedge, she's the head of research at Kite Pharma. She also serves in the board of directors of Adaptive Immune Therapeutics, in the scientific advisory board of Bolt Therapeutics, and is a founder of Genex Bio, a pre-seed venture in women's health. Most recently, Priti was the chief scientific officer at Foundation Medicine, where she led the strategy for molecular diagnostics product development for patients with cancer. And prior to that, she was at Genentech for 10 years, where she led the translational science for cancer immunotherapy, drug development, and where she, I imagine she connected with our other 2 instructors, and she's hopefully enjoying her visit today to the campus at Genentech. Priti has a bachelor's from Bombay University in Advanced Pharmaceutical Sciences, a PhD in pharmacology from SUNY Buffalo, and she also did postdoctoral research in human genomics, the Institute for Genomics Research. Thanks for joining us, Priti. And our third instructor is Rhea Nersesian. Rhea has been an instructor in this course before, as well as Heleen. She's the Senior Companion Diagnostics Project Leader at Genentech. She was at Roche for, I think this might be your whole career, 30 years. How many sabbaticals is that? I can't even count.
Yeah, only one, because it's only Genentech. Roche Diagnostics didn't count.
Oh, okay, good information to have. And during this 30-year tenure, she held various positions in the diagnostics division, including R&D, patent law, and regulatory affairs. She's part of the companion and clinical diagnostics team, where she supports diagnostic development in both oncology and non-oncology programs. And she began her career at Thurs Corporation. Oh my goodness, I have forgotten about this. I thought, and this is amazing, where she contributed to early development of PCR. And she has a bachelor's in genetics from UC Berkeley and a master's in biotechnology and innovation in regulatory science from Purdue University in Illinois, right? Mm-hmm. Wonderful. So without any further ado, Heleen, take it away.
Yes, thank you. So welcome everyone. Thank you for joining. I'm gonna start this presentation, but as Dolo said, we're the three of us can chime in, and we were really welcome questions throughout and have this more as a dialogue. The rough layout of the biomarker session today is here. I'll do a brief intro on biomarkers. Then Priti and myself will talk about PD specifically, which is really important in drug development. We have a section on predictive biomarkers, which Rhea will take on, as well as companion and complementary diagnostics, prognostic and safety biomarkers as well. And then we'll end with a really fun quiz and some case studies. All right, so really key before we dive into our biomarker work is really important to be aware that language matters in biomarker world. There's a lot of terminology and people use them they mix them and not always at the right reason or the same reason so different people talk about the same thing using different terminology. And so the guidance we like to give is to be as specific as possible when you talk about biomarkers. Don't talk about a biomarker, but really talk about what type of biomarker, what is the intended use, what is the impact of that biomarker. And so that's a kind of a key takeaway from this course. We're gonna really explain what the different things of terminology means. Starting with the definition of a biomarker. A biomarker is actually an official definition that was established through a workshop of the FDA and the NIH. It's a characteristic that is objectively measured and evaluated as an indicator of normal biological or pathological processes or pharmacological responses to a therapeutic intervention. So it's really anything that you can measure quantitatively, ideally, and it tells you about the biology of the patient or an intervention. And why are biomarkers so important in clinical development? In this course, you will learn about the different functions and the different roles of the different groups in phase, as molecules progress from discovery all the way through launch and the different clinical phases. And every part of this is really important for the successful development, as you will hear. But really key is that biomarkers play a key role here. And they really help inform decision-making at different stages of development. Drug development is very challenging, very costly. The overall success rate is pretty poor. And so it's really important that we make, efficient decisions and really informed decisions. And this is where biomarkers play a key role. Some of the analysis that has been done, the whole pharma industry has lots of experience with successful and also unsuccessful drug developments. And when people do retrospective analysis, and this is not even the most recent one, this is from a couple of years ago, looking at the recent, one of the key reasons for failures in phase 3 was really but this quote from these authors here is, the way to improve phase 3 success rates is to avoid wishful thinking and to rely on high quality scientific evidence by fully testing the mechanism against each target indication, using well-defined endpoints and in the right patient populations in phase 2 trials. So basically the key takeaway is, by the time we move molecules into phase 3, we're really not sure about a lot of things yet. We're not sure about the dose. We're not sure about the molecule hitting the target. We're not really sure that we have the right patients. And so biomarkers can really help address these questions early on so that the phase 3 will have a bigger success. So there really are 4 reasons why drugs may fail. You might have the wrong target. So you block the target, but it really doesn't result in any clinical benefit, or you have a safety issue that is associated with blocking that target. You could have the wrong molecule. So you might not actually, you might dose it, but you might not have sufficient coverage, not sufficient exposure. Maybe it's not potent enough. And so the target might still be valid, but the molecule needs improvement. Could also look at the wrong outcomes. You know, there's not a good understanding between the target and the clinical outcome that we have, as well as the wrong patients. And Priti will speak to this, or we will touch base on this on the companion diagnostics and sections later on. But, you know, really within a given disease, different biological pathways drive the diseases, and so it's really important that you enroll the right patients. And so biomarkers really have the potential to to inform, you know, each of these 4, and hopefully each of these 4 reasons why drugs may fail, and we really try to avoid failures, increase the success rate. And so we'll share more of that in the beginning. So let's, I mentioned this workshop, the Biomarker Working Group, which is referred to as BEST. This is really our biomarker Bible. You can Google it, and it's always updated. It was like jointly put together, like I said, with FDA's and NIH, but also Pharma and has been able to comment on it. And they really describe what the different biomarker categories are. And some of them are listed on this slide, and I'll briefly touch base on some of these definitions here. First of all is susceptibility or risk biomarker. It's a biomarker that indicates the potential for developing a disease or a medical condition in an individual who does not currently have clinically apparent disease or a medical condition. So it really is, if you measure this, you can predict if a patient is gonna develop the disease. A monitoring biomarker is a biomarker that is measured serially to assess the status of a disease or a medical condition or the effect of a drug over time. And so that is what you wanna measure if you wanna see if a drug works or if a patient is progressing in disease. A safety biomarker is a biomarker that is measured before or after an exposure to a medical product or an environmental agent to indicate the likelihood, presence, or extent of toxicity as an adverse event. I think that one is fairly straightforward. As a definition, not always straightforward to find them for novel therapeutics. Moving on, one more slide of definitions. A pharmacodynamic biomarker, this is like a key part of our presentation today, is a biomarker that is used to show that a biological response has occurred in an individual who has been exposed to a medical product or an environmental agent. A predictive biomarker is a biomarker used to identify individuals who are more likely than similar individuals without the biomarker to experience a favorable or unfavorable effects from exposure to a medical product or an environmental agent. So this biomarker really identifies patients who are more likely to respond than others to a given therapeutic. This one really gets confused often with a prognostic biomarker, which is really a biomarker that I use to identify the likelihood of a clinical event, disease recurrence or progression in patients who have the disease. And so a prognostic biomarker predicts disease. A predictive biomarker predicts response to a therapeutic. That is one way to keep them separate. Now, I have to say, one particular biomarker can fall into all of these categories. It can be both prognostic, it can be predictive, it can be dynamic and a response to treatment. And so when you're talking about a biomarker, again, it really is important that you talk about how you're going to use it in the context of your clinical studies. And so these three, I put them on purpose on the same slide. We call them the 3 Ps. PPP, pharmacodynamic, predictive, and prognostic. These are ones that we use a lot in drug development. And so that's, again, the focus of our talk. So moving into the pharmacodynamic biomarker, yeah.
Could you use an example of a biomarker that could predict all 3?
That could do all 3. We have one now. NFL in multiple sclerosis, it's prognostic of disease progression.
So patients who have that biomarker fare poorly.
Yes, and then they also are more likely to respond to high therapeutic efficacy agents, and it also goes down upon treatment. Yeah. Okay, are there any questions from the audience? This was kind of like the dry definitions can be kind of dry, but it really is important that we, set the stage. And so we'll have some more scientific examples in the next section. So intended uses of PD biomarkers is really, we're trying to characterize drug, target, what is the biological response that would ultimately lead to efficacy. And so we try to characterize, so that you can see there's different levels going from the drug to the efficacy. We can refer to them as a PD, as a target engagement biomarker. Like, does the drug bind the target? Does binding to the target, blocking the target, stimulating the target result in the expected biological response? We refer to that as pathway biomarkers. Sometimes people refer to them as mechanistic biomarkers. Sometimes people refer to them as target proximal biomarkers. Again, there's different terminology for this. And then there are biomarkers that are kind of linked to efficacy. And then people refer to those as disease PD biomarkers or pathophysiology biomarkers, sometimes distal biomarkers. And so they really are used to confirm targets. Do we have the intended biological effect? And can we assess our confidence of efficacy? And again, we'll have some examples there. So the target is really specific to the target. It's like that is target engagement. You measure the target, and it's really important to make sure that you can say, like, did we dose sufficiently? Did we get drug to the site of action where the target is expressed? Then, like I said, we want to make sure that blocking the target actually leads to the expected biological effect. And then ideally, if we have a good enough understanding of target and disease biology, there might be biomarkers that will allow us to give some indications that this target will result into, blocking this target will result into efficacy.
And just one point to add with target engagement is it's absolutely essential to know if your drug is hitting target, and it works for both large molecules and small molecules. So an example of a small molecule is if you're developing an AKT inhibitor, for example, the change or reduction in phospho-AKT in peripheral blood could be a measure of target engagement.
Yep. And it is essential, to Priti's point. It's not always easy. Working in the neuroscience space, sometimes your target is expressed in the brain. Can't really measure that unless there's an imaging. Besides imaging, there's very little, we don't have access to brain tissue. And so what we then try to do is use as proximal as possible PD pathway biomarkers that you can measure outside of the brain. to really then to infer target engagement. But what that will require is for us to really understand the biology between target and the biological response, which we try to do in the preclinical space. And so I'll try to touch base on that in some of the examples. So key questions that really PD biomarkers aim to answer: does dosing and subsequent exposure to the therapeutic result in target engagement? Does target engagement result in the expected downstream pharmacology? And do we have mechanistic evidence of inhibition of the biological pathway? And then is this a mechanism linked to the clinical outcome that we're interested in? And then do we understand the relationship between exposure, safety, and efficacy so that we can define a therapeutic window and stay within it? Sometimes we don't always have the luxury to just dose more and more, then you run into safety issues. Okay, the first example I'm gonna describe to you is this anti-beta-7 antibody that was in development for inflammatory bowel disease. We refer to it as etrolizumab. And beta-7 is an integrin that is expressed on, I have to put on my glasses to see my own side, sorry, on epithelial cells, on lymphocytes, and it binds to its partner E-cadherin in epithelial cells and plays a role in keeping inflammatory cells in the gut. So this is a schematic of the gut with the lymphocytes in the tissue. And so alpha E beta-7 is one integrin. It keeps the cells in the epithelial, and the other integrin is alpha 4 beta-7, which ensures that the cells leave the blood vessel and go into the tissue. So blocking beta-7 is expected to disrupt both of these. So it's preventing lymphocytes from infiltrating the tissue, and it also is expected to block the retention in the epithelial cells. And so what did we do for biomarkers for this project? So we're starting with the preclinical space. And so in mouse models of IBD, this was one in a skid mouse model of inflammation. It was shown that if we treat these animals, and so this is a model where we transfer cells into a mouse model that don't have these T cells. When we block with the anti-beta-7 integrin, you can see that these cells are not trafficking to the gut. Is this? I hope people can see my pointer. The beta-7 blocked treated animals, they don't have these cells infiltrating in the gut compared to the healthy control. And this blocking of cell trafficking was specific for the colon, because in the spleen, beta-7 doesn't play a role in migration to the spleen. And so this really showed that anti-beta-7 does indeed play a role, prevent trafficking to the gut. So beta-7 is a good target in the mouse. That was the first step. Then thinking about how can we measure target engagement? And so we use these preclinical models to actually measure receptor occupancy. So the beauty is these lymphocytes traffic through the blood. And so you can measure beta-7 by flow cytometry. And so a receptor occupancy assay tries to look at the receptor on the lymphocytes, and if the drug is bound, your detection antibody won't see it. So you need a competing antibody for this assay. And so what our data found here is in a cynomolgus study that this shows you the PK and the PD with exposure of the drug. So this is for individual animals. So the bars, the lines on the top as the drug exposure. And then the lines at the bottom are the receptor occupancy. And so you can see that as there was a lot of drug in all 4 animals, there was like complete receptor occupancy all the way through the dosing. And as the drug cleared, so as it was getting removed from the systemic circulation, you can see we were able to detect beta-7 cells on these lymphocytes in the blood again. So what this allowed us to do is really pick a dose that would result in a certain exposure that would then result in a full receptor occupancy, because our goal was we need full receptor occupancy to have the maximum effect of our anti-beta-7 antibody in these patients. Then we transferred, that was kind of the goal, the objective of our phase 1. We did our phase 1 and we wanted to see, obviously it's safety and tolerability is a key objective for phase 1, but then for biomarker was at what dose do we maintain receptor occupancy in the blood? And so what you can see here for the single ascending dose, so one dose with increasing doses, this is on the left, you have the drug exposure, and on the right, you have the receptor occupancy. And you can see the placebo stays like here flat in the open circles, there's no receptor occupancy. And then with all doses, we kind of achieved near complete receptor occupancy, but then you can see that the recovery as the drug cleared is really dose dependent. And so this allowed us to narrow down the doses that we needed to take into phase 2. And so then then there was, but then this is in the blood. And so we also really wanted to know how much tissue occupancy, receptor occupancy do we have in the tissue. And so we incorporated that in our phase 2 study. And so in our phase 2 study, we picked 2 doses, 100 and 300, based on that phase 1 results. And again, in the blood, so we can see here, we have full receptor occupancy throughout the dosing period. And then we were able to take biopsy from the gut and look at receptor occupancy in the tissue. And we were also in some subset of patients able to show that compared to placebo in the black here, with both doses, we did see compared to screening, so pre-dose at week 6 and week 10, that there was receptor occupancy at both these doses. And so this is another key point to be mindful of. Measuring target engagement in the blood is typically the easiest, but you really do need to understand how that translates to your site of action in the tissue. And so oftentimes we use the preclinical models to establish that link, because we're not always lucky enough that we're in the position to collect tissue, but if we can, we will do that. So that's targets. Then we wanted to see do we have any evidence that blocking beta-7 will actually result in the biological effect that we expect. And so of the same biopsies that we collected, RNA expression was measured, and I'm showing you here some of the cytokines that were shown to be inhibited with our beta-7 antibodies. And this is expressed by patients that were either considered remitters or non-remitters, which is one of the clinical endpoints. So like people who had a clinical benefit did indeed show reduction in expression of these key cytokines who are known pathogenic cytokines in IBD. So that really, helped us elucidate a bit of started to elucidate a bit of the mechanism of action of beta-7.
One thing to point out here is oftentimes when you enter into a phase 1 study and you propose investigating these biomarkers, your clinical teams or your leadership might ask, what fold change should we expect to see? And oftentimes it's really hard to translate murine or preclinical pharmacodynamics to human. And what I mean by that is if you see a 50% reduction in your biomarker in preclinical models, you wouldn't expect to, as a result, see 50% reduction in humans. And so that's just, a thing to keep in mind when you're looking at mechanism-based biomarkers, is they tend to be directional versus being quantitative. And it's the holistic amount of data that investigates all of these together that helps you make a decision on whether the drug is behaving in the manner you would expect it to behave or not. Would you want to expand on that?
No, I think that's a really good point. Maybe, and so that's from a mechanism perspective. I think another key question, and it's not addressed in the slide, but we can discuss it a little bit, is this question on how much, like specifically for the target engagement, how much target engagement is needed for efficacy? And so that's also a really hard question to answer. So we try to do that preclinically, through dose ranging, sometimes using knockouts and heads, and see, like, if you modulate preclinically the target only half, does that result in full efficacy or not? Oftentimes in immunology, or like in the area that my team supports, we try to maximize target occupancy. So we try to go for 100% blockade if we can safely do. But there are some targets where safety is an issue, and where we have preclinical evidence that 50% of target inhibition might be sufficient. And so when you want to use target engagement as a go, no go early on in clinical development, it's really, it's setting that bar, like what is the expected effect size on the biomarker is key. But it is a difficult discussion to have, because we often, struggle translating preclinical to clinical. It gets really easy when not really easy. It gets somewhat easier if you have a backup molecule, or like if it's already a validated target with a drug, and then you can inform like next generation of a similar class of molecules.
And to that point, Heleen mentioned this in one of the first slides, is having a tool that's very well analytically validated allows you to then make these quantitative decisions with higher precision and higher confidence, versus going in with something you developed in the lab that you don't have much confidence in, testing it in humans, and realizing that you can't interpret your data at all.
Yep. Did you want to say something?
No. I just, as far as like the companion diagnostics perspective on that, it's all about validation levels. Right. So we'll get into that a little bit later.
Yep. And so this just summarizes the atezolizumab example, where we really used the preclinical study to establish that PK-PD relationship and develop a translatable PD biomarker, like the receptor occupancy was really from preclinical species, mouse, cyno, to people. Then we used the phase 1 to establish it in healthy volunteers, actually, to establish the PK-PD relationship. Pick the dose that would then be moved into phase 2, and that's where we started to looking at efficacy. And so it really supported the dose selection, and it increased the probability of success, technical success, that's what PTS means, for our subsequent studies. One more example, another example, which really speaks to, maybe addresses some of the point that Priti just made about the totality of the data. This is an example of a molecule that was in development for rheumatoid arthritis, anti-lymphotoxin alpha, paticlizumab. It's no longer in development at this point. Lymphotoxin alpha is expressed on B cells and T cells, and it plays a role depicted here. It has many different receptors and interactions with other cells in the immune system, in the synovial fluid. So binding of the antibody directly to the cells can delete these cells. It can hold on, my pointer. It disrupts this binding to macrophages and block downstream signaling here, as well as here. And so, But all of this happens in the synovium of our, diseased joints. And so how do we go about establishing a biomarker for this particular molecule when you can't get to the site of action? And yeah, so it was really hard. We weren't able to measure a target engagement for this one. And so what the biomarker scientists did is actually, okay, like if we disrupt these pathways or if we deplete these cells, what are some of the downstream pathways that we would expect? And so those are highlighted here in red, and those are the ones that we were measuring in the blood. And so speaking again to the totality of the data, what we found in our phase 2, I'm showing you here first one of them, CXCL13. We saw that one of these pathways that we had really established as a key marker for blocking lymphotoxin alpha, we saw in the phase 2, I'm showing you the phase 2 data, a really nice reduction for this drug, CXCL13. And our drug was the red line, and this was a head-to-head to our competitor, to a standard of care, adalimumab, which is a TNF-alpha inhibitor. And so you can see that both of these drugs blocked CXCL13 similar level compared to placebo. So this confirmed that our drug was active and it was having the same biological effect as anti-TNF. So nice, but now what? Then we looked at all these other biomarkers, and so here is just depicted as a heat map. And so CXCL13 for our molecule was really the strongest change compared to placebo. And if you then compare that to adalimumab, you can see that the anti-TNF really had a much stronger biological effect on just the biology as we had known it for rheumatoid arthritis, playing upon a role in RA. And so based on this really supported our decision not to move forward with our drug, our anti-lymphotoxin alpha, because it did not have nearly as great of an effect as an anti-TNF, which was supported by the actual clinical outcome, which is depicted here. ACR20, ACR50, and ACR70 is the clinical readout in arthritis. And the blue is the anti-TNF, and it did have greater benefit than the red. But in a phase 2, there's a lot of noise in this type of clinical data. Sometimes it's not so obvious. People might argue maybe this is good enough, but having that mechanistic data showing that an anti-TNF really had much stronger effect on the totality of the biological data really made us confident that we didn't want to move forward with this anti-lymphotoxin alpha data. Okay. So those were, I have one more example before I hand it over to Priti. Those were examples where we measured either the target or downstream pathway in the blood, which is something we do a lot. But I alluded to in the beginning, sometimes our targets are not that accessible. And so I used an example in the brain, but this is an example of a target that is expressed on sensory neurons. And it's called this TRPA channel. So that's expressed on sensory nerves, and it gets activated through different ways, and stimulation of this calcium channels leads to potentiation of the sensory nerves, which results into coughing. And so we're interested in it for some of our respiratory diseases like asthma and COPD and maybe even chronic cough. Stimulation of this channel also, releases other mediators like substance P, neurokinin A, which then contributes to the inflammation that we see in these diseases, as well as they can directly result into smooth muscle contractility. And so thinking again about our target, a calcium channel on a sensory nerve in the lung. So you're a scientist, you get this project, and the team wants to know, like, how do we dose this? How much do we need to block this sodium channel, and how can we measure this? And so what the team did is, this was not an easy question, but what the team took advantage of is that sensory nerves are everywhere, and they're also in the skin, which is a very accessible tissue. And so we in our early development studies in phase 1, we leveraged the fact that we said that we can stimulate this channel by applying this irritant AITC on the skin that will result in a little bit of a redness and swelling. And you can see here imaging images of that that is taken with this laser spectral contrast imaging tool. And here you can see a picture of a volunteer where we put a little bit of the AITC there. And then, so this is a way to stimulate the AITC, the TRPA channel, and we use that to assess target engagement and pharmacological activity of our small molecule. This is a small molecule inhibitor. We did that preclinically in several models, and I'm showing you here preclinical. So it was also a model that was really nicely transferred from preclinical to the clinic. And here is, again, really beautiful data in phase 1, where we have increasing doses here with the different colors. And what is showing you here, the red bar, that was the bar that the team said we wanted to achieve based on PK and the preclinical PK/PD. On the right is the inhibition of the dermal blood flow. And you can see with increasing doses, like the highest dose is the purple bar, we were able to completely prevent that dermal blood flow, that swelling, that redness on the skin. And so this allowed us to then pick the doses to go into phase 2. Now, the key question in phase 2 is like, how does this skin translate to the lung? And what can we measure in the lung? We're doing those studies right now. I don't have any data to share with you on that one. I did actually share that we did it in preclinical animals. And so what, the totality of the data really show on the left here is really the PK/PD relationship in preclinically and how that translates to the human phase 1. So that's really nice to set what exposure we need. I think this ends my section. The examples that I've shown is really how you can use target engagements and pathway biomarkers to support go, no go, like we did for the anti-lymphotoxin alpha, to inform dosing, like we did for TRPA and beta-7, and therefore increase the probability of success. Now, my role is all outside of oncology. Priti has a lot of experience in oncology, and so there's maybe some subtleties, and she has more experience, one thing with effective drugs, and which allow you to do more things. So I'm going to pass it on to Priti, and she will take it from here. Yes, we should ask questions. Does anybody have any questions on my section before I move it to Priti?
Okay, so well, there is one question in Zoom, so we can start there. Mariana asks, can you comment on Project Optimus and the role of biomarkers in dose optimization?
Yeah, so Project Optimus is an initiative from the FDA. It's very focused mostly in oncology. Maybe I don't know how familiar Priti is with it. But yeah. So you want to speak to that?
So what the FDA has done, let me just back up first. Traditionally in drug development, at least in oncology, clinical trials are designed to hit the maximum tolerated dose. So the way in which you design a phase 1 study, it's called a 3+3 dosing regimen, where you have these dose escalation studies and 3 patients in each cohort. And with every next cohort, you have an up, you scale up your dose to the next level. And you keep doing this until you hit toxicity. Now, as a result, many of the drugs that have been approved in oncology have a fairly, sizable toxicity profile associated with them. Now, many of these drugs are much more effective. I shouldn't say much more. They are effective at doses that may not be as toxic. But drug developers have really shied away from going forward into phase 2 trials with a biologically effective dose, but rather they go with a maximum tolerated dose. And so what the FDA decided to do was put this initiative forward called Project Optimus, where the onus is on the drug developer to demonstrate why they picked the dose that they picked for a phase 2 trial, to the extent where drug developers, the FDA is asking drug developers to run randomized trials in phase 2 and 3 with multiple doses if you show to them that you're moving forward with a maximum tolerated dose. And this is where showing a biologically active dose becomes really important. And the FDA has, in fact, asked drug developers to incorporate biologically active dose finding in their phase 1 and phase 2 trials. And that's essentially what Project Optimus is. And it actually goes very nicely into this next segment, but I can touch upon it when I discuss the categories of pharmacodynamic markers. Maybe we can just go to some more questions.
Yeah, I was going to add, I think it was really specifically for ONC, because in the indications that my team support, we never dose. It is always about what is the minimum dose that we need to see efficacy, like that. So it was really a different approach to dosing.
Safety is paramount in immunology, because cancer patients have no other options. It's not right to keep dosing to maximum tolerated dose. So it's a good initiative, I think, by the FDA, but it makes understanding biology all the more important, and doing this work that we're talking about here all the more important.
Another question, Nathan?
Yes. Next question. Can you, in general, use PD biomarkers to narrow your therapeutic doses?
Yes.
Okay. And then next question. Can you please clarify terminology around PD biomarkers and proof of activity? I've often heard them used interchangeably.
Yeah, that's a very good question. So proof of activity is probably the goal of the trial. So a PD biomarker can be used to provide evidence of proof of activity, but proof of activity itself is not a biomarker, right? So proof of activity is like the objective of a trial. That's the way how I describe it.
Yeah. I think there's two ways of looking at activity. One is biological activity, and the other is clinical activity. And they're two very different things. You can have a drug that's biologically very active, but it may not translate to clinical activity. And clinical activity at the end of the day is the gold standard. That's what we're all here for, is to identify drugs that are clinically active. So there is, it comes back to that first slide that Heleen showed on the terminologies used. Oftentimes when you're sitting in a room, you can be debating about apples and oranges and not realize you're debating about apples and oranges because you're using the same terminology. So activity in this instance could really mean two things. Are you talking about biological activity or clinical?
Okay, and then this next question's a little bit long, so it has kind of two questions in it. So some oncology clinical trials enroll as many patients as possible, especially when no other treatments are available. Do you have any comments on how to balance getting favorable clinical trial outcomes with compassionate drug use, even if such patients may not be on the right ones, such as biomarker expression? And additionally, how much impact, if any, does the study design considerations have on the FDA's decision to approve or not approve a drug?
I'll address the first question. When you design a clinical trial, you have to get that trial protocol approved by the FDA and approved by the institution's IRBs. And both the FDA and the institution's IRBs look for what was the data package that led to you deciding you want to treat patients in that specific disease. So even in the instance where, you know, you're thinking of a compassionate use approach, you need to have a really clear scientific rationale as to why you're going into that patient population. So without that, the FDA wouldn't approve your protocol, and more importantly, the institution's IRBs wouldn't approve your protocol. To the second question on how important is clinical trial design, pharmaceutical R&D is all about the experiment in humans. The clinical trial design is the most important aspect of drugs getting approved and commercially viable. And I'll just give you an example of that that I came across just recently, where there was a cell therapy developer that they were the first ones to develop the cell therapy. And they were so careful in the way they designed their clinical trial that they made sure that patients were made to stay in the hospital for at least a week while the cell therapy is, after the cell therapy is infused back into the patients. And the reason why they wanted to do that is they just didn't know if the patient goes home and something catastrophic happens and it's bad for the patient, it's bad for the company, and so on and so forth. So that's how the trial was designed. And ultimately, the label of that drug, the drug got approved. The label said this can only be given inpatient, meaning if you get on this drug, you need to make sure the hospital has a bed for a week for that patient. A second drug developer came in and said, we're going to design our clinical trial such that patient comes in, gets their drug, and they can go home. Okay. And if they fall ill, they can always call the hospital and come back. And that difference in the way the two clinical trials were designed resulted in that second drug not getting that on the label that it has to be inpatient, meaning it could be outpatient, which makes it easier on the patient, makes it easier on the clinician, and actually makes it easier for that drug to be clinically commercially viable. So the way in which clinical trials are designed are fundamentally important for getting a drug approved and for making that drug commercially viable. I think after the MOA of the drug and understanding who your patient population is, the third most important thing is the design of the clinical trial.
Okay, and then I think we have two more questions, and then we can move on so that we're keeping with time. So the one more is, can you discuss optimizing favorable biomarker results and side effects?
Can you optimize variable? Is that what you said?
Favorable. Can you discuss optimizing favorable biomarker results and side effects?
Not sure I completely understand the question, but
I think that what they're asking is, can you use biomarkers? I'm going to reframe the question. Can you use biomarkers to, I guess, identify a dose that gives you a better side effect profile? Is that
yeah, I see somebody now.
Okay.
Yeah. And so yes, you can. You know, if again, like if you know the biological effect, the wanted biological effect, if you can measure that, and then if you have your safety liability, and you know, it kind of comes down to the threshold, what kind of effect do you want, you know, and how much safety liability can you tolerate? But yeah, that is exactly how you would use biomarkers.
Can I ask another on that? Would you still go with one dose in phase 3, or would you still try 2 doses?
Oh, that totally depends. Okay. That depends on so many things. The reason we go with more than 1 dose in the clinic is if we haven't nailed it down. Like oftentimes the regulators want us to go as low as we can. And so if we haven't shown like a no effect dose, then we are stuck with testing 2 doses. And if there's no safety issue, we can push it as high as possible. Ideally, we want to stick with 1 dose because it's just way costly to do another dose, and studies studies take too long.
Okay, and then last question for now. Can you please comment on what I3+3 clinical design means?
That's for Priti. Are you going to
Yeah.
speak to that here or not?
No, I wasn't
Yeah.
planning on speaking to it here, but a 3+3 clinical trial design is you have what's called step-up dosing in a phase 1 trial in oncology, where you start at a dose level that is minimally active, okay? So you start at a dose level that is barely tickling your biological pathway. That's your first dose level. The second dose level will be an order of magnitude higher. And again, you enroll 3 patients. You wait for a month after that those patients have been dosed to address safety. And if they're safe, you now can open a third cohort, which has an order of magnitude higher dose. And you keep doing that until you've hit a maximum tolerated dose. So a 3+3 trial design is where you have step increases in doses in each cohort, and you enroll 3 patients in each cohort to understand safety in a given window, and that window is usually 1 month.
Okay. I think we got to move to your
Yeah.
section.
Okay.
Yeah.
Do you want to sit here or you
Yeah, I
want
can
to? Okay.
So what I thought I'd do here is, Heleen really nicely framed the concept of pharmacodynamic biomarkers. I wanted to use a slightly different flavor in how you can think about pharmacodynamic biomarkers. And many of the principles are very similar to what Heleen described, and she's given many examples of those as well. So like I mentioned earlier, biological activity is different from clinical activity. And that's essentially the point here, is one category of pharmacodynamic biomarkers is to address, is this drug biologically active? But a really important point here is when you look at a biomarker, when you look at changes in biomarkers to address biological activity, they may not be associated with clinical efficacy. In fact, sometimes they have nothing to do with clinical efficacy. And I'll give you 2 examples of what we mean by pharmacodynamic biomarkers that fall under that umbrella of biological biomarkers. The first one is what Heleen pointed out as well, is does my drug hit target? She gave a good example of receptor occupancy for large molecule inhibitors. There's also copies of infused product per microgram of PBMC DNA for cell therapies. These are really good examples of drug hits target. These can be assessed systemically in blood. So, you can get your result for drug hits target from every patient, and they're often used for PK/PD modeling to understand, am I seeing a good correlation between the dose in that 3+3 study design and the drug actually hitting the target that's intended to hit. Another example I gave you earlier was with an AKT inhibitor and looking at changes in phospho-AKT over time in peripheral blood cells. You can use that data to do PK/PD modeling and assess, is my body reacting to my drug in the way I expect it to? And like I mentioned, these can be observed in all patients, okay? The second one of example of a biological biomarker is mechanism of action related. Now, this is really I would say the cornerstone of pharmacodynamic biomarkers in oncology. When you develop a drug in oncology, everyone wants to see this. Is your drug doing what it's intended to do in humans? The reason being, and I'm sure it's the same thing in immunology, is mouse models don't really represent human disease biology. And so when you're developing these drugs in humans, it's really important to know, am I doing anything at all? An example here is tumor infiltration of CD8 positive T cells, change in pathway signaling in the tumor. Now, what's really important for mechanism of action related biomarkers is it requires the site of action context. So for drug hits target that I mentioned earlier, you can measure this biology in blood in a non-invasive manner. For mechanism of action related biomarkers, it's really important to study that biology in that disease context, okay? So in this case, it's tumor. It may not be dose related. This is really important. Biology in every patient is different, okay? So you can imagine it's like the volume knob on a radio. In some cancer patients, that volume knob might be really high. In some other cancer patients, that volume may be dialed down to an extent where you come in with your drug and it doesn't do anything. So there is a lot of heterogeneity and variability in patients. And so understanding a dose response for a mechanism of action related biomarkers is a tall order, and I can't give a single example where that has worked out. The third point here is these mechanism of action related biomarkers may not be observed in all patients. Again, to the point I just brought up earlier is phase 1 trials tend to be done in all comer patients, okay? So you can have a colorectal cancer patient, a breast cancer patient, a pancreatic cancer patient, all part of your phase 1 trial. Each one of these patients has a really different disease biology. Your pathway biology is really different. You don't expect your mechanism of action related biomarker to behave similarly. So that really adds to complexity in observing a mechanism of action related biomarker in your phase 1 dose escalation trial. Oftentimes for MOA related biomarkers, what we tend to do is we tend to reserve this only at the dose expansion cohort. What is a dose expansion cohort is when you go through your phase 1 trial for, 3+3 trial design, you hit a dose where your clinical scientist says, that is my maximum tolerated dose. That's the dose I want to take forward in a phase 2 trial. Now let's go from just 3 patients in that last dose to 10 patients. That dose expansion cohort is the best place for you to study mechanism of action related biomarkers. It's 1 dose level. Generally, those patients tend to be of 1 disease type. So you have less heterogeneity that's disease to disease based, less heterogeneity in terms of dose, and it gives you the highest chance of actually looking at biology. And so that's where mechanism of action related biomarkers tend to be studied, is in that dose expansion cohort at the last maximum tolerated dose. Now, Project Optimus is starting to question a lot of these, and what clinical trialists are now doing is expanding at 2 different doses. At a dose that they think is biologically active, if they see responses with the drug, then hey, let's expand that dose. And at a dose that's got maximum tolerated, a dose stamp on it. So if you now expand at 2 different doses, you can actually use your MOA related biomarker to define what is my biologically active dose. A key point here is that this MOA related dose may not be associated with efficacy, and I can't stress that enough, that none of these pharmacodynamic biomarkers have anything to do with clinical efficacy. So you can't really de-risk an asset in a phase 1 study based on MOA related biomarkers, unless you don't see it at all.
So that's what I mean. Unless it is a negative, right? Like if it's not sufficient, but it's required to see some biology.
Yeah.
So you can, like in OMNI, we would use it to, or in outside of oncology, we use it to refine our dose. Like the doses that you see nothing, it still informs, you know, how to move things forward.
And I'll give you guys an example of where an MOA related biomarker helped with a no-go decision. This was in a cancer immunology program called an anti-OX40 program at Genentech. And in preclinical mouse models, it was really clear that we saw Treg depletion, okay? Regulatory T cells are depleted in tumors. And that was our pre-specified hypothesis in the clinical trial protocol. In the protocol, we wrote that we had to see depletion of T regulatory cells. And when you put it in the protocol, there is more validity to your hypothesis than you sitting in your lab and assuming, oh, let me just study this and convince people. You can't convince people if it's not in the protocol. It has to be a pre-thought experimental design and a pre-thought hypothesis that you're now following up in a clinical trial. We recruited 20 patients in our expansion cohort, all of whom provided pre-post biopsies. We had a really well-validated assay to look at T regulatory cells. And with very high confidence, we were able to say that, oh, guess what? We don't see Treg depletion at all in human tumors. And it turned out that OX40 levels in human Tregs are much lower than mice. And so we had this exaggerated pharmacodynamic effect in mice that just didn't translate to humans. And we ended up killing the program in phase 1. But it took that investment of enrolling 20 patients at an MTD and doing the biopsy study to get to that point. So this can be an expensive endeavor.
Could you have done, in hindsight, could you have collected some tumor samples and looked at OX40 expression ahead of time?
We did that, but
Okay.
you know how it is.
I don't know what I just unplugged. I'm not gonna hold you.
And you know, the way it is in drug development is for your leadership to be convinced, they have to see.
Yeah.
The proof is in the data.
Somebody is talking while not on mute. We're getting some feedback. Thank you.
Any questions on biological pharmacodynamic biomarkers? A second category of pharmacodynamic biomarkers is what I call clinical. So the first one was biological, the second one is clinical. Clinical biomarkers are the ones where a dynamic change in the biomarker is very clearly associated with clinical efficacy. This is considered the holy grail pharmacodynamic biomarker. Examples of clinical biomarkers include changes in tumor markers. Example, I don't know if you know what CA125 is. It is a cancer antigen that's expressed on the cell surface of every ovarian cancer tumor, and it's shed in plasma. And is in fact one of those examples that Heleen mentioned. It's a prognostic biomarker. It can also predict clinical efficacy, and it's a pharmacodynamic biomarker. It's used often in ovarian cancer clinical trials to determine if a drug is doing something and as a clinical endpoint in clinical trials. Prostate serum PSA is a second example, a prostate-specific antigen in prostate cancer. And a third example is a change in circulating tumor DNA, and I'll speak to this a little bit in subsequent slides. And of course, the last one is radiologic imaging, which can also be a pharmacodynamic biomarker, but is also very clearly a clinical biomarker and an endpoint in clinical trials. Now, the nice thing about these biomarkers is that they can be evaluated non-invasively. They have to be, because you have to detect them in every patient. You can't get a biopsy in every patient. Many patients are, it's not easy to surgically biopsy every patient. So these markers have to be evaluated systemically. They confirm the clinical translation of biological pharmacodynamics, meaning if you showed that my MOA changes, and guess what? It results in a radiologic change in efficacy, or it results in reduction in circulating tumor DNA. That is your best clinical biomarker package that you can share with your clinical team. One point that's really important is these biomarkers are unequivocally related to outcomes. You cannot use a novel thing in your phase 1 study and say, I know this is going to be my clinical biomarker. You need a lot of legacy data to inform what that biomarker needs to be. And, all the things I've mentioned here, there's a lot of energy now, both at the level of the FDA and at various cancer consortiums, to really utilize circulating tumor DNA as one of those biomarkers that has an unequivocal relationship to outcomes. This requires a high quality regulatory grade tool that you will hear about later in the context of companion diagnostics. And this may be employed as an endpoint in clinical trials. So this is an instance where you need to know what that quantitative magnitude of change is. When Heleen showed an example of IL-17 earlier, and we talked about, we don't know if a 50% reduction is good enough. In this instance, when it's a clinical diagnostic with a very clear association as a regulatory endpoint, you have to define quantitatively what that change is. I am looking for a 30% change in radiologic imaging. I am looking for a 30% change in circulating tumor DNA, and use that in your statistical analysis plan in the design of your clinical trial. Okay. So I'm just going to give you, Heleen gave a few examples of drug hits target. I thought I'll just give you a couple of examples on MOA-related biomarkers. An example here is in cancer immunotherapy. This is with a drug called atezolizumab, which is a checkpoint inhibitor, and it blocks the PD-1, PD-L1 interactions that enables your T cells, surveilling T cells, to actually recognize tumor cells and kill them. And this is an example where we had a post-dose biopsy. This was a renal cell carcinoma patient. We had a pre-dose biopsy rather, where you can see the brown dots that represent CD8 positive T cells. This patient, we received a 4-week on-treatment biopsy that's in the bottom here. And this 4-week on-treatment biopsy, we looked at CD8 positive T cells again. And what we observed was a dramatic increase in the number of CD8 positive T cells in the post-dose biopsy compared to the pre-dose biopsy. Now, this is what we call an MOA-related biomarker. It's not directly pathway associated, but you know that if you're blocking PD-1, PD-L1 interactions, it should now allow your T cells to see tumor cells. So you should see an influx of T cells into your tumor, and that's exactly what we saw here. And then we also employed gene expression to show that, in fact, these T cells that are entering into the tumor cells are activated. You're seeing a hyper, interferon gamma signaling pathway that's upregulated in these tumors. So the green bars here are the pre-dose biopsies. The light green or khaki color bars are gene expression changes in the post-dose biopsies, and you're seeing an increase in gamma interferon and granzymes and perforin and so on and so forth. So it tells you that not only are the T cells coming in, but they're activated and charged and ready to go out and kill the tumor cells. And lo and behold, this patient at 3 months started to show a response to the drug and started to, survive for a good 3 years after, progression-free survived for 3 years after the initial dose of the drug. So this is an example of an MOA-related drug. What's nice about MOA-related biomarkers is that they can also help you understand response and resistance patterns in tumors. And this is from the same phase 1 study where, we had pre-post biopsies from pretty much every patient in this trial. And we observed that in patients who do not respond to this therapy, their baseline biopsies that are shown above here, these are 3 distinct patients. The baseline biopsies had very few CD8 positive T cells to begin with, or they had no T cells at all, that we called immunological ignorance, or they had some T cells, but they were really sparse compared to the example I showed you earlier, or the T cells are present, but they're present in the stromal interface and do not enter the tumor. And when you treat these patients with atezolizumab, you can see more T cells in this instance here, but they're still stuck in the periphery. They're not entering the tumor, and those patients didn't respond to the drug. In this instance, in the middle, where there are few T cells at baseline, there continue to be few T cells post-dose. And in the instance where there are no T cells at baseline, there are no T cells post-dose. So this really helped us understand why these patients don't respond to the drug. And they don't respond to the drug because they just don't have enough T cells, or the T cells are not in the right place in the tumor microenvironment to essentially mount an effective immune response. And that ultimately helped us understand what causes resistance to these drugs. And we came up with, this concept of a tumor immunity continuum, where in fact there are 3 types of tumor microenvironments. The highly inflamed tumors that I showed you in that renal cancer example earlier, where these patients respond really well to immunotherapies. They have a lot of T cells in their microenvironment and do very well. The second example is what we call immune excluded tumors, where T cells are present, but just not in the right place. And you can add enough drug on these patients and it does nothing to them. But also the underlying biology below that is very well defined here. We took a lot of time to understand what are the biological signals that are associated with these tumor patterns. And then, of course, these immune deserts that tend to have no T cells at all, do nothing. They tend to be highly proliferating and have a lot of fatty acid metabolism signatures associated with them, where the tumor cells are just gobbling up all the energy and not letting any energy go to the immune cells in the periphery, and just as a result, killing the immune cells. So this is an example where MOA-related biology can actually help you understand a lot about the disease, help you understand a lot about the drug, and helps you actually build a pipeline in the future for what your next generation assets need to be. So a little investment that goes into MOA-related biomarkers can reap a lot of rewards in the future. And that's just, it comes to a value proposition statement when you invest so much in this, in this space. The last example I'll give you here is PD biomarkers correlated with clinical outcomes. And an example I shared with you is circulating tumor DNA that's really rising to the top as a really good surrogate of clinical activity. And I'll just preface this by saying today, if you're looking at clinical efficacy, and overall survival tends to be the gold standard for clinical efficacy, it can take you anywhere from 3 years to 7 years to determine if your drug has clinical efficacy or not. And this is where surrogate biomarkers like circulating tumor DNA come really handy, where within 6 months, you can determine, is my drug going to have that overall survival benefit or not? And doing this early in phase 1 and phase 2 trials helps you understand, should you invest in a phase 3 trial or not? Wait 3 years to do that? Perhaps not. You can get an answer in a year. And an example I'll show you here is what we call increasing speed to signal finding. The blue lines here, the y-axis is change in tumor volume. Okay, the x-axis is time in months. Each bar here represents a single patient's tumor dynamics. And in this instance, with this drug, most of the patients showed a change in tumor volume by 2 months based on radiographic imaging. But when you look at the change in circulating tumor DNA that's shown in blue, within 3 weeks, you can show that, hey, guess what? Circulating tumor DNA is completely abolished in these patients. You're getting complete clearance of circulating tumor DNA by 3 weeks. When you see such a pattern of complete clearance of circulating tumor DNA, it gives you a really high confidence that this drug is going to show great overall survival benefit. Okay, there's enough data now in literature to show that clearance of circulating tumor DNA correlates with overall survival. And this can be used as a surrogate for you to make decisions of moving from phase 1 to phase 2. And in fact, even using circulating tumor DNA as a surrogate endpoint in phase 2. So you can get your result in 1 year instead of waiting 3 to 5 years.
Priti, can I have a question on this? Really fascinating. Are there any plans to potentially move this into an approvable biomarker for accelerated approval or something along those lines?
Yeah, absolutely. The second example I have here was actually where the company worked with the FDA for making ctDNA an approvable endpoint. But the FDA is working very closely with drug developers and diagnostic developers to make ctDNA an approvable endpoint. It is going to be disease specific. It may also be therapeutic modality specific. So there's that aspect that needs dialogue between the FDA and the drug developers, but it's absolutely already an endpoint in hematologic malignancies, and it's now moving into an endpoint in solid tumors as well.
Thank you.
So this is an example of a drug called tebentafusp, which was a GP100 vaccine that was developed in uveal melanomas. Uveal melanoma is a really deadly disease. There's been really no drug that's been approved in this disease. And this drug had 0% response rate, okay? Not a single patient responded to this drug. The progression-free survival hazards ratio for this drug was 1, meaning the patients on this drug progressed at the same rate as placebo. But the overall survival hazards ratio for this drug was 0.5, which was really shocking. And this is really the first example of a drug that has no impact on progression-free survival and has a dramatic impact on overall survival and not a single response rate. What this company did was they said, hey, let's look at changes in circulating tumor DNA. And if circulating tumor DNA is a better endpoint than radiologic imaging, remember in oncology, for progression-free survival, radiographic imaging is the gold standard. And that was what was used. And there was no change in radiographic imaging. And what this company saw was that indeed, when they use circulating tumor DNA, over 50% of their patients showed a reduction in circulating tumor DNA, and that's shown in this graph here, by I believe by 6 weeks to 9 weeks after starting tebentafusp treatment. And that reduction in circulating tumor DNA shown in blue correlated with an improvement in survival in these patients versus the bar shown in green, which is this green and this black here together, okay? And what they did, which was really nice on this bar here, was they said, let's look at the various changes in circulating tumor DNA that's on the x-axis and associated impact on overall survival on the y-axis. And what they saw was that patients who had the greatest reduction in circulating tumor DNA, that means clearance of circulating tumor DNA, had an overall survival hazards ratio of close to 0.1. And this was why this drug ultimately got approved. And today, this drug has rapidly become the standard in uveal melanoma, despite the fact that when you get on this drug, you have no idea if this drug is working or not. To me, this is really remarkable that you get on this drug, nothing happens to you from a response perspective, but you show an improvement in survival, and it's now become the standard.
Is anything known about the mechanism?
What's believed is when you have inflammation in the tumor, radiologic imaging, which is a CT scan or an MRI, cannot, your tumor size sometimes remains the same.
Okay.
Although the tumor has actually shrunk. But because of all the inflammation, there's no impact on tumor volume.
Got it.
And so ctDNA now becomes a much more precise marker of tumor reduction than imaging.
Okay.
And this is my last example, and we can shut it off from here. But this is an example I wanted to show where circulating tumor DNA is used for dose finding. Okay, this is an EGFR inhibitor in non-small cell lung cancer that addresses these two mutations, T790M and C797S. And what we did here was we looked at those two specific mutations and looked at clearance of those two specific mutations in a dose response study with this company, Arvinas. And what we saw was that what you're looking at here, these different colors, let's look at this here. At different colors represent the different dose levels. 25 mg is in purple, 50 mg is in green, and so on and so forth. And what the company saw was that at 25 mg, there's no clearance. And in fact, they didn't even see responses. So that is a very minimum dose that's not active, very clearly. You now start to see activity at the green bar here. Okay, but activity starts to improve as your doses increase. If you look at 200 mg, your reduction in ctDNA is a bit all over the place. Now, when you hit 400 mg, remember, this is a 3+3 dose escalation, right? So you're seeing 3 patients in purple, 3 patients in other colors, and so on. Green and blue, unfortunately, we didn't have enough circulating tumor DNA in the baseline sample to monitor change. But when you have enough circulating tumor DNA in the baseline sample, for example, in this 400 mg dose, 2 out of the 3 patients showed complete clearance. Now you can use this biomarker and go for Project Optimus to the FDA and say, I'm going to go forward with 400 mg per kg because 2 out of my 3 patients showed complete clearance in ctDNA in the biomarker that is designed for that drug. Okay, so this is a really nice example of circulating tumor DNA used for dose finding, and in fact, using it with your FDA regulators in justifying your dose to move forward to phase 2 and phase 3. So that was really a quick walkthrough for examples of biomarkers, whether it's mechanism of action or clinical, to inform decisions, inform science and biology and target discovery and clinical decision making. Any questions?
Thank you, Priti. There are questions in the Slido. I don't know if someone, Heleen, wants to read them. I'm happy to read them.
No, we're working off the same computer, so I don't.
Let me read you some of the questions in the
Okay.
Slido, starting from the most recent ones. One question from Jessica. Do drugs like Taben, Taben, with an unknown MOA have a more difficult pathway to approval, or is showing clinical improvement, in this case, overall survival, sufficient?
It's generally very difficult if the mechanism of action is unknown. The FDA likes to see that in their package. And the reason being, when your mechanism of action is unknown and you hit a safety event, you don't even know why you hit that safety event, okay? It could be on target, it could be off target, but if you don't understand that, it's hard to disconnect safety from efficacy. Now, if you decide to go ahead and do it anyway, and you happen to be lucky and you see overall survival benefit, I don't know what Taven is, and I haven't come across an example like this where mechanism of action is unknown.
I know.
Yeah.
And so it really depends on the effect size, and also if there is already options, treatment options for patients. Pirfenidone for IPF, we have no idea, despite it being around for 50 years, how it works. And it got approved. It had marginal efficacy, but at that point, there was nothing else for these patients. It's a very deadly disease. So most patients die, within 5 years. And so that's an example of a drug getting approved despite us, nobody understanding how it works. So, but it really depends on, is the other option, what is the unmet medical need, and how big is the clinical benefit?
That's a great example.
Yeah.
That's great. Another recent one, the Amilex drug for ALS. I think.
Yeah. So again, like a very severe, debilitating, deadly disease. So, at some point, the FDA, the regulators, they're also hopeful. We don't know why, but if it helps patients, let's go for it.
Wonderful. Another question from Greg. Could you comment on the role of in vivo imaging biomarkers in clinical trials? When is it appropriate to use PET, CT, MRI versus blood biopsy biomarkers?
Do you want to talk about Alzheimer's?
Yeah, I mean, it's appropriate every time it answers the key question. So if the key question is target engagement, or the key question, so again, like what is the intended use? And so sometimes imaging is the only way you can get to your answer. It absolutely is appropriate. Now, it is expensive. They're not always without risk for patients, and so there's lots of efforts to try to find surrogates that are more feasible, that, for instance, in Alzheimer's, we're trying to find blood-based biomarkers that reflects similar things, pathology that you can measure with imaging. But yeah, they're super critical when they answer the critical question, and we will incorporate them in our trials.
Yeah. To Helen's point, I'll give you an example in oncology where the thing with, you can use different radio-labeled probes. For example, you could use a radio-labeled CD8, let's use that. If you are looking for infiltration of CD8 positive T cells, you could use radiolabeled CD8. You have to go through regulators because you're injecting something into humans. So the biomarker plan needs an IND as well, and needs to demonstrate safety. To Heleen's point, it becomes really expensive. So it really depends on, if that's the only thing available, then you need to go for it. And if you have the means and the funds, go for it. But yes, it requires an IND. It requires FDA engagement to utilize imaging-based biomarkers.
Thank you so much. A question from Kumari. How do you assess clinical or practical sensitivity and specificity for PD biomarkers?
So multiple ways. So sensitivity and specificity. So the last one, specificity. A PD biomarker doesn't have to be specific.
Yeah.
And so all you need to know is, does it change if I block a pathway or I hit a target? An example is the CXCL13 for anti-lymphotoxin alpha. It was reduced by anti-TNF and with an anti-lymphotoxin alpha. It's a very useful biomarker for us to show that the drug did, what they're supposed to do at that dose. So it doesn't have to be specific. In terms of sensitive, I mean, this is what we do before we go into the clinic. Do we have sensitive enough assays to detect? Do we have sufficient window to inhibit a change? Do we know what the longitudinal variability is within a given patient? Do we know what the impact of standard of care is of all these patients? And so we tried over the years, collect cohorts of samples to actually, prior to us doing these studies, to characterize that, so we know what to expect.
And the assay that you use also you need to understand what is the performance of your assay. And so sensitivity clinically also is dependent on how robust your assay is. If your assay is just generally variable, then you better see a significantly large dynamic change for you to feel confident. And to Heleen's point, we do a lot of work on the simplest pharmacodynamic biomarker to just understand how the biomarker performs in healthy donors. What does blood shipment do? If you're moving from Siberia, a patient, at -4 degrees to 45 degrees, what does that temperature change do to your biomarker? All of those things have to be taken into account when you're trying to understand sensitivity. And to her point, specificity is not really needed in this instance.
In this for PD biomarkers don't have to be specific. It's an assumption that everybody makes. And then there's well, how do you know it's specific? And we don't. It doesn't have to be.
Yeah. A companion diagnostic definitely needs that, but a pharmacodynamic does not.
Wonderful. One final question, and I'm going to read it now. For some reason, I only see three questions in Slido, and I don't know if there is a problem with the Slido that is canceling out questions.
Oh, Dolo, sorry. So I'm moving questions to the archive.
Oh, Nathan, you're messing with it. No worries, no worries.
Yeah.
So I'm going to read this question, and then we can sort through that. And again, we also want to take a 10-minute break, and there's going to be another chance to ask questions from 1 to 1:30. So question from Mailing. In general, how long does it take to find a biomarker that is clinically meaningful, say associated with clinical outcome? Does it also go through phase 1, 2, and 3, or a few trials?
For a biomarker that is associated with clinical outcomes, and if you're intending to use it for decision-making, you need a lot of clinical data prior to using it in your trial.
I mean, the main thing you need is for your drug to be clinically active, which you don't know until you get to phase 3.
Yeah.
So it can take decades because all of our molecules, the majority of our molecules fail. And so we always assume that something is going to predict efficacy, but you need those approved drugs first.
Yeah.
So that you can establish the link between treatment response and efficacy.
I'll just give you an example of, if you want, you're developing a drug in non-small cell lung cancer. it's going to take you 5 years to hit your endpoint of overall survival. You want to use a ctDNA as a biomarker and as a clinical endpoint. You will need a cohort in non-small cell lung cancer that's separate from your trial, and really understand what does a 30% reduction in ctDNA correlate to in terms of hazards ratio for outcomes. And then use that information in your statistical analysis plan for your trial. So you need an independent cohort and have data generated with the same assay that you're using in your trial, and understand the performance and the impact on hazards ratio, and then pre-specify that in your trial.
Wow.
So
Wonderful.
yeah, it's a long journey.
Yeah. Right now it's a long journey, but as these become more and more standard, like in heme malignancies, MRD, ctDNA based MRD has now become a standard, but it took 15 years to get there.
We're talking decades.
Yeah.
Yeah.
Wow. Thanks so much. I see one more question. Nathan has allowed one more question through from Mike. Are biomarkers always necessary when treating obvious conditions like tremor or dyskinesia? And that's one question. And then there is a follow-on question of a different nature. What about when repurposing FDA-approved drugs where the receptor occupancy is well characterized?
Yeah, I can start. So yeah, sometimes things like tremors or dyskinesia are really easy to see. That's a clinical endpoint. And so what that doesn't allow you to answer the question, what if your drug doesn't work? What if the tremors stay the same? Do you understand why it fails? Did you hit the target? Did you completely block the target, but the biology just is irrelevant for the tremors? Or did you dose wrong? Was it the wrong molecule? And so that's one. What was the other question? Lost my train of thought here.
Yeah, the other question, hold on a second. I want to make sure I read it exactly as posted. What about when repurposing FDA-approved drugs?
Yeah, in other indications. Yeah, so that's a really good point. So if we're, and it doesn't even have to be an approved drug. So sometimes our molecules, you do a phase 1, you look at the receptor occupancy, for instance, like target engagement, and then you go into 3 different indications. We might go into COPD, respiratory, and cough at the same time for that TRP agonist that I said. And so it depends on how confident you are about the target expression in those different diseases. So if you know that a particular disease has a lot more of the target, you might need to dose more. But it is something that we need to characterize and think about. Sometimes, the target is not that abundant and we know we dose sufficiently. And again, I'm talking from an OMNI perspective where safety is not the biggest liability.
I guess it depends on if you're going at the same dose as the FDA approved dose, then go for it, But if you have to go dose finding again, then it becomes a whole exercise all over again.
Yeah.
So it really depends. If you're going at the same FDA approved dose, there's very little, I guess.
Yeah, I mean, it's like repurposing oncology drugs for non-oncology indications, where there's lower, tolerance for safety. We might actually do dose ranging of that dose, and we're doing that in phase 1 for some of our bispecific antibodies. Yeah.
Wonderful. So thank you so much. How do you guys feel about taking a break now before Rhea's session?
I think that sounds perfect. That's what we were planning for.
Fantastic. So it's 11:42. Let's come back at 10 minutes exactly at 11:52. That wasn't hard math. 11:52.
Yes.
Wonderful.
That's good.
Thank you.
Thank you.
All right. Welcome, everyone. I see our instructors are ready to continue. So Rhea, take it away.
All right, we're back. We're back from our break and ready to move on here. So we're moving on to a predictive biomarker, and just coming off all the really great discussion we had in the first section, just remind everybody that with the predictive biomarker, me coming from a companion diagnostics perspective, I'm really thinking about biomarkers at baseline. So this is when a patient presents with a disease, They haven't been treated yet. So these are the biomarkers we're looking at to decide, you know, how their journey might be in our trial or with a drug. So from that perspective, a reminder about a predictive biomarker. So we're using it in this case to identify individuals who are more likely than others that don't have the biomarker to experience either a favorable or unfavorable effect. And so this is predicting patients who might respond better to a drug. This can be applied both in a efficacy as well as a safety perspective. And this is what we would call patient selection. So the term intended use, this is a very important term with all of our biomarkers. When somebody from one of my molecule teams comes to me with an idea of how they want to use their biomarker, my first question is, what is your intended use? And intended use also has a regulatory meaning, but it also helps me to determine, you know, are we talking predictive, are we talking prognostic, so that we can have the right discussion. So in this case, you know, an intended use of a predictive biomarker is, as we've just described, is really in this concept of personalized healthcare, where we're aiming to identify a subgroup of patients that would have an enhanced benefit or perhaps an enhanced risk for taking this drug. So really the vision of personalized healthcare is the diagnostics and the biomarker diagnostics together with our pharma partners, working together to find the right treatment for the patient, meaning not every treatment is right for every person, and the diagnostic is what we're using to determine that. So this is really a PHC, or personalized healthcare. And one of our most big examples here is with Zelboraf. About half of melanomas carry a mutant BRAF gene. Vemurafenib, or Zelboraf, it's a BRAF inhibitor that inhibits the altered BRAF protein, and it's approved for patients that have a BRAF mutant melanoma. So as you can see here, before treatment, the patient was experiencing severe symptoms, and after 15 weeks on Zelboraf, had an amazing response. So underlying this, yes, so also to say that patients don't always continue in this path. Unfortunately, sometimes the patient may become resistant to the drug, and symptoms may return. So from a CDX point of view, we're looking at this intended use of the drug and the intended use of the diagnostic in a formal way in the label of the drug and in the label of the diagnostic. Here, where Zelboraf is defined as a kinase inhibitor for treatment of patients that have a BRAF V600E mutation as detected by an FDA-approved test. So all that is very specific. So if you do not have this BRAF mutation, you are not prescribed Zelboraf. So the V600E mutation status is the essential decision maker here for determining if this is the right drug for you. And this is a companion diagnostic. This is a classic companion diagnostic. And you can see in the Kaplan-Meier curve, the separation of the population from the treated Zelboraf patients on the top, and I think that's a standard of care below. You can see a clear separation in the curves. So the companion diagnostic product that resulted from this work is the COBOS mutation test. Perhaps many of you are familiar with the Roche COBOS line of, in this case, a PCR-based product. So this is an assay that runs to detect alterations at the V600 location. It's a real-time PCR test, and it has been FDA approved for use together with Zelboraf. there have been a history of diagnostics CDX approvals. It actually goes back to Herceptin. When we think about the HER2-ish assay, it's way back in 1998. That was actually a companion diagnostic in the making before the regulations and everything were developed around the concept. Since that time, we then moved through the EGFR work, the Zelbaroff example I gave you. We move into Tecentriq, where we talked about PD-L1, IHC. We talked about that a little in the first session. And up through November or the end of 2017 with the approval of Alecensa with the detection of an ALK fusion positive type of NSCLC. So all of these examples that we've talked about, it's pretty straightforward, especially the Zelbaroff example. There's one mutation that leads to very severe disease. So the path for developing a CDX and the path for the drug development go hand in hand together down a pretty straight path. We know what we need to do. We carry the drug through appropriate phase 3 trials. We engage with the regulators, but in the end, we always know where we're headed. Of course, the complexity of the landscape in the CDX world has really evolved, and no longer are we looking so much at those single gene alterations. We're looking at a much broader landscape. And certainly that's true in oncology. In the OMNI or non-oncology space, this is especially true because there are so many factors behind a patient's disease. It's not just a single gene that's being affected. So from a CDX perspective, we have to navigate these twists and turns and guide our programs to take logical risks in developing a CDX, but not to overdevelop a box like the Zelbaroff COBOS assay. That's a box that's manufactured with shrink wrapped and shipped around globally. That comes with a very high cost to develop. We can't be developing every single biomarker in that way. It's not cost effective for us or for society, really. So we've had to look for new solutions. So backing up a little to the actual definition of some of these from a regulatory perspective. So this again is a US regulatory perspective, which is now moving more into the EU, which is a little outside the scope of our talk today. But if any of you would like to talk to me about CDX evolving in the EU space under IVDR, I'm happy for you to reach out to me later. So companion diagnostic, as we've seen, it has that official definition of it being a medical device that is essential for the safe and effective use of a biological product. This is a restricted label, meaning the drug can only be used in a patient in this subgroup of patients. So we don't always want a restricted label. A lot of times we want our molecules to be for all comers. So a CDX does divide and restrict the labeling. But of course, it does reach the patients that are most likely to benefit. Note that a CDX is actually a medical device, which isn't necessarily just a molecular biomarker target, kind of like we've been talking about today. A medical device could also be something like an algorithm or an imaging algorithm. We could get pretty creative of where companion diagnostic could be defined. You may have heard this alternative term called a complementary diagnostic. This is the case where the data is very important in it could aid the physician in deciding what treatment to give their patient. So it's clinically meaningful risk-benefit data. However, the drug is effective in all comers. So the label of the drug will be labeled for all comers and it will not be restricted in the way we talked about for CDX. I will note, however, that this definition of complementary diagnostic is a bit of a lay term. It's not clearly defined in regulations. It has been seen employed in oncology space, and I'll give you an example of that. But it is still a very gray area.
Can I ask a question?
Yes,
Rhea?
please.
Do you think this will evolve? Like, how do you think this will evolve? Do you think we're gonna get more complementary diagnostics, or do you think that we tried that and it's not that valuable and we might not use them going forward?
Right. I have in oncology, we've seen examples where the complementary diagnostic test being it's not required to prescribe the drug, the test wasn't quite utilized in the way that I think was initially envisioned, because physicians could prescribe the drug for their patient without taking the test. So why should we develop the test? However, outside of oncology, where diseases are more complex, I can see a complementary diagnostic, or whatever, maybe we give it a different term, could be very useful for physicians in selecting the patient's treatment. Yeah.
Okay. That's good.
But we have yet to
Got some
see
work
those
to do.
examples.
We've got some work to do.
We do have some work to do. So this is an example of a drug and a diagnostic developed together, early days of Tecentriq, atezolizumab, and the PD-L1 story. And the initial internal plans were to have this be a companion diagnostic. So the PD-L1 test for Tecentriq in the case of bladder cancer was designed and internally designed to be a CDX, straight up. However, when the data read out, it was clear that there's efficacy in the PD-L1 low as well as PD-L1 high. And if you can see the numbers, they're a little bit small, but the overall response rate in PD-L1 low is lower than it is in the higher expression, but there is still response. And in this case, it's the FDA who decided that this is not a companion diagnostic because this should be offered to all patients. So I'm going to pause here because I often get some questions about this to see if anybody wants to ask me before we move on.
Yes, Rhea, we have one question for your section so far. Do complementary diagnostics cover diagnostics that measure adverse events that often occur with a given drug and would require stopping using the drug?
No, so here again, we're talking about baseline determination. This is my brain is solely focused on baseline biomarkers and not monitoring in this case. But as we said, biomarkers can fall into all of the categories. And certainly if in the trial we see there can be adverse events and monitoring is necessary and the patient may change their treatment, that would probably be it would raise to a high level of regulatory oversight. I don't know, it would be necessarily a companion or a complementary terminology.
Maybe just to clarify, I'm not sure if everybody clarify the term baseline. Baseline is before treatment starts, just so people understand what Rhea means with baseline. So yeah, this is all treatment
before
you start treating.
Yes. All right, any other questions, Nathan?
None right now for this section.
All right. Okay. So what about predictive biomarkers outside of oncology? So the companion diagnostic field and the FDA guidances that you could Google and read for your leisure are all developed around oncology. So we ask, what about outside of oncology where the space is more complicated? And there are predictive biomarkers in asthma, as we see here, but these do not rise to the level of companion diagnostics because they were already existing as a clinical diagnostic test. So these were not tests developed specifically for the drug to measure efficacy and safety of that particular drug. This is part of the disease diagnosis. So this was not labeled as a companion or complementary diagnostic.
Yeah, so I like to always speak to this example because it's clear, and again, coming back to the terminology, despite outside of oncology, there not being companion or complementary diagnostic tests, there are examples of predictive biomarkers. So it really distinguishes like a biomarker versus a test. Like a companion diagnostic isn't a biomarker. A companion diagnostic is a device to measure a predictive biomarker.
Yes.
And like people always use that. They think a companion diagnostic is a biomarker. It's not, it's a test. And so again, like terminology matters, although I get accused of being the word police in my team.
Yes. Yeah, that's why I like to show that box. Like if you think a companion diagnostic is a medical device manufactured into a box and shrink wrapped and shipped around the world. That's the companion diagnostic that we're talking about. It's run in routine labs. The guts of it is the biomarker. Yeah. Thank you. So the pathway here is the same. The regulatory pathway in the US is the same, whether it's a companion or a complementary diagnostic. The level of safety and efficacy and data and validation of the assay and all of that is the same for either companion or complementary. The diagnostic submission pathway goes into the CDRH division in parallel with the drug submission. So the one of the roles of my team in a companion diagnostics role is to ensure these timelines are in parallel, because the approval of the diagnostic and the approval of the drug need to come out contemporaneously, which practically means within plus or minus 30 days. So it's a pretty tight window we need to reach. Yeah. So we have learned over time that the different divisions of the FDA don't necessarily communicate pieces of things to each other. So we as the sponsor company together with our diagnostic partner have to work very carefully to make sure both divisions of the FDA are staying informed during the process. So a few considerations. So as in the example I gave of the early PD-L1 Atezo, we had the company had a plan for it to be a companion diagnostic, but the FDA re-looked at the data and determined it was complementary. And that is a reminder to everyone that yes, you can decide, oh, I want to have an all-comer drug, but we want to have a test. So we're going to have it be complementary. But in the end, the FDA will review the data and they will determine that labeling. So yes, you can have a plan. You can go to the FDA and have a pre-submission meeting. You can discuss, but until the trial, the phase 3 trial is complete, you're not going to know for sure if it's a companion or a complementary designation. Another interesting consideration. Oh yeah, go ahead, Priti.
Would you want to share the cost of developing these drugs, whether it's a companion?
Of the tests?
Yeah.
Well, the cost depends on the platform and depends on if you're working with a new platform and so let's just say something like mass spec. Your costs are going to be quite considerable because you're going to have a lot of validation to do, not only of the instrument platform itself, but the biomarker and the assay as well. If you're working on a well-established platform, let's say you're working with Ventana on an IHC with a stain, the costs will be less because the infrastructure
less because they might not know what
the less. So let's say maybe with I don't want to quote this, it'd be millions of dollars and up. $1 million would say it would be a low cost companion diagnostic.
It's a very expensive endeavor is what I was trying.
It is very expensive.
It's never $1 million.
It's always way more.
Yeah. Several million for the cheapest. And I think if you develop a whole new platform, that's like triple-digit millions.
Yes.
Yeah.
And again, the validation and the costs for companion versus complementary are exactly the same. There's no shortcut.
Rhea, I have a question. Would companies typically try to position a diagnostic as a complementary versus a companion?
Yes, they may come in with the position that they feel the drug is appropriate for an all-comer population, but yet the diagnostic offers significant value to the physician in determining whether to prescribe the drug or not.
There have been talks of how can you use the data with the complementary diagnostic to perhaps put your pricing strategy together. So there's so much, especially when you're looking at a global registration of a drug, some countries will not pay you if your outcomes aren't a certain X. And the complementary diagnostic really helps in negotiating pricing with different countries where their expectation of benefit to pay is different from the United States. And one thing to point out here is, the path that you take for companion complementary with the FDA is also very similar with the European health authorities, for example, and it can help you during price negotiations.
Right, so yeah, I've just put a couple things for thought. Yes, that's exactly right, Priti. The reimbursement is an important discussion to have from a commercial perspective. So for example, if the diagnostic test is not required for prescribing the drug, perhaps the test will not be reimbursed at all. Or as Priti said, some countries may utilize any diagnostic test to sort of turn it in, take a complementary and turn it to a companion from the aspect of reimbursement. So these are all things more just food for thought for you to kind of as you process this whole topic. So yes, there are many challenges around when and why a physician would order a complementary diagnostic test. I can tell you with Atezo bladder cancer example, our supply chain was geared up to sell many PD-L1 tests, but when it became designated as complementary, they did not sell as many as predicted, because physicians saw an opportunity to put all of their patients straight on the drug without a test. So we've been talking about this one indication, one drug, one test, one allele, that straight line on the path. But really, as we move into drug development today, we're building this pyramid where we're moving towards multiple tests. For each patient, there could be multiple biomarkers for which we want to test for. And when we're in oncology, we may have a limited amount of tumor tissue. We have perhaps a limited amount of testing that can be done. So this moves us in on this pyramid of complexity to the bottom level, where we're looking at an NGS-based biomarker test, where we can screen for multiple genes and multiple things in one test. So this is the push for the need for this consolidation of diagnostic testing and not one shrink wrap box for each individual biomarker. That's not really going to work as we learn more and more about these complex diseases. So just a quick refresher, and this is meant of something you guys can print out and refer to, that there are kind of 3 buckets of predictive biomarkers: the companion, the complementary, and what you could call a standalone diagnostic or a disease diagnostic. This would be where I would put the example we talked about with eosinophils for asthma. These are diagnostic tests that are important, can be referenced in your labeling, but they're not considered a complementary or a companion diagnostic. I think in Alzheimer's space, the amyloid test also falls into this bucket of standalone diagnostic, where there's a measurement of the amyloid in the patient to determine their amyloid tangles and potential for Alzheimer's disease. That is in the standalone diagnostic space. And I'll pause again for any questions.
Yes, 2 questions. What is meant by all comers labeling?
Yes, that means that the label is not restricted to a certain population. So the label could be for patients who have pancreatic cancer. You know, and without any limitation to a specific biomarker.
Yeah.
So they wouldn't necessarily have to have PD-L1 positivity or an ALK fusion. Anything else to add?
No, that's good.
Yeah.
Okay. And how much overlap is there between the content of the CDRH diagnostic regulatory submission and associated CDER/CBER therapeutic agent regulatory submissions?
Yes, so there are certain aspects of data that are shared with both divisions, but of course, the CDER, the drug division, is going to be most interested in our drug development data, and CDRH is going to be most interested in the validation of the diagnostic test, that it's safe and reproducible and meets the claim with which we've asserted its intended use. So there is some data packages that are provided to both, but their focus is different.
We're doing okay on time. So
Okay.
I think I can zoom through the next section before giving it back to you for your example.
Great.
You want to keep presenting?
Sure, I can click
Oh,
for you.
Heleen, one question did just pop in for Rhea. Can I ask?
Okay.
Yeah, theoretical question. What happens if someone does use a drug, even if it does not have the required outcome from the companion diagnostic? Is it just that the drug will not be effective, for instance, so it's a waste of resources?
It's a reimbursement thing, you know, if you're using a drug outside the prescribed label, you may not get reimbursed for it.
Yeah, but I mean, the physician is able to do what they think is best. So, you know, a companion diagnostic may also be for safety. So it's possible if the CDX is a safety type of biomarker, they could be putting their patient at risk if they have not tested for it. But again, that's the physician to decide. It's the labeling. We put forth what we have studied. And again, the reimbursement issue could be a problem as well. I hope that answers your question.
I'll give you an example. If you have an EGFR mutated lung cancer and your drug is approved in the third line, meaning you've gone through 2 different drugs, failed, and now on third line, and your doctor goes, oh, here is a new patient, they have the same EGFR mutation, but they're second line, can I give it to them? They could, because it should theoretically work, but it's not in the label and they wouldn't get reimbursed.
Okay.
All right. Right, we'll pass it to Heleen now.
Yeah, this next section about prognostic and safety biomarkers is higher level. So I'll do quick because then we have a cool example before we move into the quiz. And so prognostic biomarker, again, is a biomarker that identifies the likelihood of a clinical event, disease recurrence or progression. And so as an example here, schematically, you can see that in the green dotted line, a biomarker positive patients have a greater decline in their health, whatever the outcome is, than the biomarker negative. And so why are these biomarkers important? I have a really great example on the next slide, which I touched upon this. They're really critical for how we decide how to help support trial design. To Priti's earlier point, like clinical trial design is so critical. It really makes or breaks your ability to test your hypothesis and your drug. And so this is an example of two phase 3 trials of pirfenidone, the drug that is approved for idiopathic pulmonary fibrosis, IPF. And you can see on the y-axis is the clinical endpoint, which is a % FVC predicted. That is your lung capacity, basically. And you can see the light line is the placebo. So over the course of several months, 70 months, you can see that the lung capacity declines in the placebo-treated patients. Now, what you can see when you compare these two phase 3 trials, which were identical trials, you can see that there is kind of a difference between the two placebo arms. So in one trial, the placebo arm was greater, the patients were more severe, they progressed faster. And the other trial on the right, it was significantly less progression in the placebo arm. And the impact of that is if you then test your drug, and again, same identical trial, pirfenidone, same dose, you can see that on the left, the company was not our company. The company was able to show a statistically significant difference benefit of pirfenidone, but on the right, it didn't. And so it's an example to show that like ensuring that you have a good understanding of disease progression allows you to design the trial in the most optimum way, so that you understand the variability, you have an expected treatment effect, how many patients you enroll, because otherwise you might not see any benefit. This is an excess. So what happened with this drug? Based on this, again, in the EU, they got approval, or the other way around, actually, I forgot. And either in the EU, they got approval, or in the US, but then the other side, they made the company do a third phase 3 to confirm. I can't remember what the FDA said or the EMA. And so it has a huge impact. And so, and specifically, you wonder, like, why were these trials so different? I can believe you, many people have tried to investigate this and we cannot figure it out. So that also shows you how complex our diseases are. And despite us doing a lot of work, clinical trials sometimes throw us for a loop, and we really need to do the best we can to understand our diseases. Okay, that was the point here. A prognostic biomarker doesn't have to be predictive of treatment effect, and this is again schematics taken from that BEST resource. It gets a little confusing, but this is just if you read through it and sit down and think through it, it will explain itself. But basically, the treatment effect can be similar in the biomarker positive and biomarker negative. So then it's really, I don't have an example off the top of my head. I don't know, do you? But it is possible. As opposed to on the next slide, prognostic biomarkers can be predictive as well for treatment benefits. And this is this shows you the same pirfenidone study, and in IPF, where again the red line is the placebo arm and the blue line is the treated arm. If we now subdivide these patients based on a biomarker, CCL18, which we had shown was prognostic, you can see that the red bar on the right is the CCL18 high patients. So, and this was just cut off in the middle of the patient. You can see that they decline much greater than the patients who are low in this biomarker, and comparing, so this is comparing the two red ones, that is the prognostic aspect. And because of that, you can see that there was actually a greater benefit in the CCL18 high compared to the CCL18 low, making CCL18 also predictive. This was done retrospectively, so not prospectively. So it's not proven. We didn't do another study to prove that it was true. So this is really us trying to understand the disease. On the next slide is another example where this time in asthma, where serum periostin was a biomarker that we were developing as a predictive biomarker for this drug anti-IL-13 that was in development for asthma. We found that periostin was prognostic for exacerbations, which is the primary endpoint in respiratory diseases like asthma. And you can see like the gray bar on the left is the periostin high patients. You can see they have a lot more exacerbations compared to the periostin low, which is the middle gray bar. And so again, that's the prognostic aspect. And then if you look at the predictive part, you compare the difference between the light gray and the blue. there's a great difference, a great treatment response of this anti-IL-13 drug in the periostin high patients, but you don't see it in the periostin low patients. And again, you can see how like this would greatly impact your trial design. If you don't use a biomarker, the treatment effect would be kind of the average, as you see on the third one. Whereas if we're able to select really on the patients with the highest IL-13 driven disease, was our hypothesis at that point, you get treatment effects like on the left two bars. The next slides we can kind of skip, but it's just for your reference because I always get mixed up when I try to talk through this. But this just really spells out, you know, like there are cases where biomarkers can be prognostic, predictive, and all the different combinations that are possible.
This is one I had printed out and posted at my desk for the first few years I worked here. Yeah, to keep it straight. So it's a very handy reference.
Yes. And so that really summarizes the prognostic biomarkers. You know, it uses, you know, it tells you about disease progression, really important for trial design. And they can also, but the same biomarker can serve multiple purposes. Are there any questions on that one?
I think this is a new question from Alice. Would a pharmacogenomic marker be considered complementary? For example, a CYP2C19 gene variant in clopidogrel, I think that's what it means.
That's a question for Rhea.
Yeah, I think so. Yes. The CYP. Yeah.
I'm not familiar with that drug.
I mean, anything that has to do with the patient responding to the drug. I mean, it could be a companion or a complementary. It would really depend on all the factors of the trial.
Got it. Thank you. And there's a question for Priti, but we can leave that for the end.
For the end. Okay.
Yeah.
All right. I just have 2 slides on safety biomarkers. Again, you know, indicate the likelihood, presence, or extent of toxicity or an adverse event. Just like the PD biomarkers, safety biomarkers can be subcategorized in the next slide based on their intended use. So they can be used to monitor a safety outcome. They can be used to predict a safety outcome. And they can also be used to predict the likelihood or the risk of an outcome. So that's again like a baseline, a pre-dose biomarker. And so intended use is important. To decide whether something is a safety biomarker, can be monitored or predictive, again, requires, when we're developing novel therapeutic, in my experience, outside of oncology, love to hear Priti's thought, it requires a lot of data in order for these biomarkers to truly predict safety. You know, there's some common things like, you know, kidney and liver function, you know, those are very well established as safety biomarkers, but novel safety biomarkers for novel targets, classes of targets, the way that it really clicked with me when I started in this field, we're trying to find biomarkers that predict efficacy. Efficacy is what we're going for, and is much more, ideally, much more prevalent than safety. Our safety events are more rare than our efficacy event, which make it very difficult to actually establish novel clinical safety biomarkers. Yeah. Did you want to add to that?
You know, the thing with safety biomarkers is oftentimes, at least in oncology, patients are on multiple drugs, you know, they're on your drug plus chemotherapy, and it becomes really hard to ascribe a safety event to a biomarker for safety to your drug. It's not that trivial to do. They're often on co-medications, you know, these patients are on other pain medications, this, that, and the other. So safety is a lot more challenging than efficacy.
Yeah. Now, having said that, the industry recognizes the need for it, and the way there's a lot of efforts in consortium type work where different pharmas, different organizations come together, you know, share the data and to really establish these novel safety biomarkers. But again, it's a very lengthy process. Okay, that's all I had on that one. And then I have one more last slide, really honing in on, we all touched base on the intended use of the biomarker and how important it is, like how you're going to use this biomarker. Priti touched base a little bit on the analytical validation of the assay. So there's the clinical validation of the biomarker. It's like, what does it actually tell you? Is it pharmacodynamic? Is it predictive? Is it prognostic? But then there is the analytical validation of the assay, the robustness, all of that. And so the level of analytical validation that is required, the intended use really drives how much validation needs to happen. As clearly, if it's a surrogate endpoint, so it's depicted here in different grays, that requires us to be the most rigorous analytically, like there's no doubt about that. Versus if we're looking at things like target engagement, dose selection, those things are really more at the sponsor's risk. We're not really actually, we're still in developing these drugs. We're not making treatment decisions. We're just interpreting our clinical trials results. And so depending on how you use your biomarkers, that really drives the validation. And so when people talk about a validated biomarker, do they mean analytical, or do they mean clinical, or do they need both? And so again, it depends on like, what is the intended use? Is all of that clear? And I'll leave it at that.
Yeah, so I can say from the CDX perspective, no matter whatever C it is, from the companion diagnostic perspective, we traditionally, wouldn't be developing that medical device until phase 3, till the data is supporting that investment. However, even early as phase 1, if the drug development team wants to use a diagnostic test for patient selection in a very early phase of the trial, that will also still require a high level of validation, and that validation will have to start earlier. So here where the CDX appears, it's really more about that development of the medical device, but the use of it in a predictive way with a high level of validation can come honestly at really any stage here.
I think we can move to your BFAST example.
Okay, so this is a fun part. I get to tell you about this really cool trial that Roche Genentech is sponsoring. A lot of these results are published, so we can really talk about it. It's called BFAST, or the Blood First Assay Screening Trial. And the whole concept here is that this is an oncology. It is a biomarker-centered trial. So the first step in a patient's journey, a patient with non-small cell lung cancer, their first step is to have their biomarker measured. And this biomarker is measured in the blood in this case. Traditionally, the biomarkers are measured in the tissue. And as we've seen from our presentation today, you can see a lot of things when you look at a slice of tissue. However, not all patients are healthy enough for a biopsy, or perhaps they don't have enough tissue left. So this BFAST study was designed to study prospectively using circulating tumor DNA to determine if a patient is eligible for treatment in the trial. So as I alluded to, many patients are never actually molecularly diagnosed in NSCLC. This is an older slide, but I'm afraid this still continues to this day. I don't know, I have examples in my family, maybe you do as well, where your family or friend wasn't even offered any molecular testing. And that's, there's many reasons for that, with which we could have a whole lecture. But one of the reasons why is that there isn't tissue available. There are many other reasons. Being able to look in the blood as a blood biopsy opens up a whole new avenue for patients that who perhaps aren't able to have a surgical resection of DNA. The circulating tumor DNA, what we've talked about already today. In this case, we're talking about that baseline or prior to treatment. This is not for monitoring. This is when the patient first presents with non-small cell lung cancer. And we can look to see if that tumor is shedding DNA into the blood, and we can look to see if that tumor has any molecular alterations. And then based on that alteration, we can decide what treatment the patient could be eligible for. So the challenges in traditional lung cancer treatment, patients don't have enough tissue. The pyramid that we talked about the numbers of biomarkers that we want to test, perhaps they've already been tested for a few. They were negative in that case, and they don't have any tissue left for testing. Some of these alterations are also quite rare that we're looking for, and we can't have one box for every single rare potential biomarker. So BFAST here offers that solution of expanding testing to a blood-based test. We're using a next generation sequencing, so it's a multiplex testing platform that looks for over 300 genes. The patient doesn't have to have any tissue available, and the trial itself is very economical to run. Our partner here is Foundation Medicine, who's now been acquired by Roche since 2018. And many of you may be familiar with Foundation Medicine, but they're a leading provider of comprehensive genomic profiling in cancer.
Somebody's very familiar with Foundation Medicine.
I know, we just say, if you have any questions about FMI, we just ask Priti here. So I'm giving just my soapbox version, which is good. So the product we're using from FMI is the Foundation One Liquid CDX, which detects 324 genes with all four classes of alterations. It also includes a biomarker known as tumor mutational burden and MSI, microsatellite instability. This is validated to a very high level in their CAP-CLIA lab in Cambridge, Massachusetts. It carries FDA approval and a CE IVD mark. It's also available and approved in Japan. You can't read this, but there's a list of their genes, which you can Google very easily. Their package insert is available publicly. So here's the study of BFAST. So we start on the left where patients are screened by 2 tubes of blood, and that's sent to FMI for central testing in their lab in Cambridge. And if an actionable alteration is discovered here, one that's covered in the BFAST study, which is in all those different colors, the patient will then be advanced to specific screening for that molecule to make sure they meet the rest of the inclusion criteria for that molecule. So here you can see the biomarker is the very first entry, and only upon being biomarker positive do they enter the screening for the rest of the things they need to match up with. In this case, the patients, in the context of the BFAST trial, if they are not enrolled into a cohort, we also track a natural history cohort to see how these patients follow up, which is proving to be quite a very large amount of data on over 1,000 patients. So we're following their NSCLC journey. Physicians also receive a full report from FMI that includes alterations other than the ones in BFAST, so the physician can then go on to look for other treatments for that patient outside the scope of our study. So we're going to focus a little on the very first cohort, which is an ALK positive fusion cohort. This study has read out. This is in connection with Alecensa. Alecensa is a drug for ALK positive NSCLC patients who harbor an ALK fusion. The current label is tissue-based, and based on the data from BFAST, we were able to show that using blood and looking for the same ALK positive alteration, that the patients were very responsive. So in this study, you can see 87.4% of the patients had a confirmed response, an ORR. So it's a very, very high number. And we were very excited just to know that, in this case, there was roughly a little over 100 patients enrolled. Let's just say 88 patients were offered an option that they weren't able to get before because they didn't have tissue available. And so this impact of blood-based testing is real for those 88 patients, and certainly for those of us on the study. We feel really strongly that a blood-based screen is the future. And I don't work for FMI, by the way, just as a disclaimer. So yes, as the result of this, FMI was able to update their label for their F1 liquid CDX product to include the ALK fusion detection as an FDA-approved targeted therapy. So in summary, BFAST has really illustrated the power of a collaboration with Foundation Medicine in these complex diseases where there can be many, many different alterations found in a cancer patient. The blood-based liquid biopsy is going to enable many more patients access to a personalized cancer therapy. And this has been an innovative trial, which started with first-line NSCLC, which is now has expanded to other arms have been added to BFAST, and other studies have been launched. There's a study called Tapestry, which has taken BFAST and expanded it to go beyond NSCLC and its pan-tumor, and currently has 15 arms with 15 different molecules and biomarkers. So BFAST has really set the stage for the future. And then, as we learned in COVID times, we can have a drive-up window for blood testing. So, who knows where this will take us.
Also for nasal swabs.
True, nasal swab. There's all kinds of things. Yes, and PCR. Now everybody knows PCR because of COVID too. So, all right. So that's it. I'm happy to address the questions on BFAST, and I don't know, Nathan, if you want to go to the quiz first, or should we do the questions that have piled up?
Yeah, there's two quick questions. I think there's one for this section and one for Priti's first section, and then we can do the quiz. Is that okay?
Yeah.
Okay, great. So this question: are patients ever positive for more than one biomarker in F1L, and how are decisions made in those situations?
Yes, so there are patients that will have more than one actionable alteration. In the context of our clinical trial, that's triaged by prevalence. So we suggest to the PI that the patient is recommended for the cohort with the lowest prevalence. And for us, that means it's the hardest to find. But ultimately it's up to the PI to decide if there are two actionable cohorts. Perhaps they feel that one molecule has a better safety profile, or other factors in the inclusion criteria that make that patient a better match for the other cohort. But our recommendation in our protocol is to go first with the biomarker with the lowest prevalence.
I'll just add that when you have two biomarkers that are positive in a patient's tumor sample, the variant allele frequency also matters. If you have a higher variant allele frequency for one of the mutations, then it's a higher likelihood that the patient might derive better benefit from a therapy that addresses that mutation.
Thanks. And Priti, can you comment on ctDNA for neuro-oncological indications, given the difficulty of rebiopsy?
That's a good question. ctDNA in neuro-oncological diseases is really hard. We haven't been able to detect circulating tumor DNA in patients with GBM, not for the lack of trying. It's just perhaps blood is not the right vehicle to assess circulating tumor DNA for GBM, for oncological diseases.
We ever tried looking at CSF?
We have not, but our hunch is CSF would be the appropriate vehicle.
Okay, great. There are another question or two that came in, but we can go ahead and maybe do the quiz first, and then we can return to Q&A.
Okay, I think last time we did the quiz, should I just go through the slides?
Yeah, you go. You can.
Okay.
It's your quiz.
Yeah,
I think we can skip this one.
yeah.
This is a summary.
Yeah, so Rhea, what we can do, I've programmed the quiz options in Slido, but I couldn't fit the whole question, so we can display the question on your slide, and then the students will be able to see the options they can choose from.
Sounds good. Let's give this a go. Okay, so we're gonna play a little biomarker Jeopardy. And so the hint here is to think about the intended use. And then you'll need to decide what type of biomarker it is. So is everybody ready? So the first question, there's a subgroup of women with breast cancer who overexpress HER2 protein, and they derive clinically meaningful responses to Herceptin, an anti-HER2 antibody. So a positive result confirming overexpression is required for patients to receive Herceptin. What type of biomarker is HER2, and what type of assay is the diagnostic?
We have about half the class, and they selected predictive biomarker and companion diagnostics.
Yay, very well done. Okay. So in this situation, PD-L1 expression greater than 5% IC is determined by a Ventana PD-L1 assay. It's associated with increased ORR in a non-randomized study of Atezo. So what type of biomarker is PD-L1, and what type of assay is the diagnostic?
Okay, we have a good number, and about 60% have answered predictive biomarker and complementary diagnostic.
Yay, 2 for 2. Excellent. All right, so this cystic fibrosis kit is a qualitative genotyping test providing information intended to be used for carrier testing in adults of reproductive age and as an aid in newborn screening and in confirmatory diagnostic testing in newborns and children. So what type of biomarker is this cystic fibrosis kit? Or is X-tag? So what type of biomarker is the genotyping, and what type of assay is the diagnostic test?
It looks like most people have selected diagnostic biomarker and standalone diagnostic.
Very good.
Yeah, you guys are good.
Yeah.
Okay, so the BRCA test detects the presence of BRCA1 and BRCA2. These mutations are responsible for the majority of hereditary breast and ovarian cancers. People with the mutation have increased risk for developing breast cancer and ovarian cancer by age 70. Mutation carriers previously diagnosed with cancer also have an increased risk. Patients with these types of mutations have increased risk. It's all increased risk. So what type of biomarker are the BRCA mutations, and what type of assay? So a hint here, you'll see nothing in this statement that mentions a drug.
Trick question.
Yeah, after you said that, Rhea, one of the options decreased in what people
There you go. Good choice. So you got to look at the totality of the statement, because there could be another intended use that does list a drug, but this particular test has a claim. These are claims taken from random FDA things I found on the internet. This is a claim here in this case without the drug.
Yeah, so yep, that's what most people selected.
Okay, so the COBOS PCR-based HIV test is an in vitro nucleic acid test for quantitation of HIV RNA in plasma. This test is intended for use in conjunction with clinical presentation and other laboratory markers of disease progress or progression for the clinical management of HIV. And that should say what type of biomarker is the HIV test. That's a typo.
And yes, about 65%, yep, have chosen monitoring biomarker and standalone diagnostic.
Yeah, this test is actually known as HIV monitor. So I took that out because that would give away the answer. This is actually a flagship test that Roche Diagnostics worked directly with the FDA in the early days of HIV. And it became kind of a parallel in COVID where Roche and other diagnostic companies worked very closely with the FDA in response to high unmet medical need. All right, this hemosyl fibrinogen test is for quantitative determination of fibrinogen based on the Klaus method in human citrated plasma on a coagulation system. So this biomarker may be used to select patients with COPD at high risk for exacerbation or for inclusion into a trial. What type of biomarker is fibrinogen?
We have it pretty mixed here. Yeah, so the most selected answer was actually susceptibility / risk biomarker. And then very closely it was prognostic biomarker.
Yeah, it's a tricky one. Yeah. Okay.
Last question.
I think this is the last question. Yeah. Creatinine, is that how you say this?
Yep.
Creatinine. Test system is a device intended to measure creatinine quantitatively in plasma and urine. The measurements are used in the diagnosis and treatment of renal disease and in monitoring renal dialysis and a calculation basis for measuring other urine analytes. What type of is creatinine?
About 50% have picked safety biomarker.
Yeah. So yes, your intuition was right. Elevated levels could signal that something is going wrong. If I got that correct. All right, so good. We're pretty much right at 1 o'clock, and I think that's the end. And everybody goes, "Yay! Nice job, people."
That's it.
Yeah, thank you. Thank you for all your attention and good questions, and we're here to answer more questions.
Yeah. Real quick, the camera in your conference room has locked onto Heleen and Priti. So Rhea, I don't know if you want to
Sorry.
scoot over.
I'm over here.
Yeah, come scoot over.
I'll just scoot over.
Yeah.
Hello.
Okay, great.
I can also stop presenting. Here, let me do that.
Okay, great. All right, yeah, we do have a few quotes. Someone did ask if we could clarify the answer for question 6 on the quiz.
Question 6.
Yeah, that one was the fibrinogen question, being a
Fibrinogen.
prognostic biomarker was the correct answer.
All right, now my computer's doing something crazy.
Yeah, so we can post the slides with the quiz answers at the end, and also the recording for the session will also have this portion also with the answers. So in one, yeah.
Let me open up question 6.
So I guess the question here was, so the answer is it's a prognostic biomarker because it is used to identify patients with COPD that are at high risk for death or exacerbation. So it really predicts disease progression. That makes it a prognostic biomarker.
Okay. So a question did I already ask the question, are patients ever positive for more than one F1L? I think that we already answered that question, yes.
Yeah.
Yeah, okay. Can you comment or speculate about the impact of the COVID pandemic on changing the biomarker landscape in a large percentage of people due to the broad tropism of the virus? For instance, it may cause cardiovascular disease that remains unnoticed or undetected, but changes biomarker expression.
So can you, how did the question start? Like, I want to try to understand whether we have biomarkers that predict the cardiovascular sequelae. Is that the question?
Yeah, can you comment or speculate about the impact of COVID on changing the biomarker landscape in a large percentage of people due to the broad tropism of the virus?
Yeah, I mean, I don't know of any example, but if I understand the question is like, if somebody has COVID, how does it impact biomarkers that we're using for some other decision making? Yeah. I mean, it comes back to the point, like, you know, when you characterize, when you have an established biomarker, you need to know, you know, the clinical variability in other indications with other drugs. And so it's a good question. Yeah. I don't think we know at this point. I don't think we have enough information. I would anticipate this would typically reports would show up where biomarkers didn't do what they were supposed to do, and people would realize, oh, that's because they have COVID, maybe.
Right.
Yeah.
As like a side note to that, mask wearing had an effect on our respiratory trials, and that with everybody wearing a mask, just regular levels of exacerbations reduced. With people staying home and being free of allergens and other things with wearing masks. So I believe our trials did see a reduced number of exacerbations, and that is challenging when exacerbation amount is one of your endpoints in reducing. So it doesn't have to do with COVID necessarily, but mask wearing did have an effect.
Okay, and then there's two more questions, but I did want to thank the students for those who are staying on. I know a lot of people had to drop off, but Dolo had to step out also. But for those who do need to leave soon, thank you all, and thank you, Heleen, Priti, and Rhea for a great session today. So our next question is, can you comment on use of AI for biomarker identification?
We're using machine learning for biomarker identification, and it's primarily used to, especially with large genomic data, to identify gene signatures that are associated with response and resistance. We've published quite a few papers on those. The second example is using machine learning from digital pathology images to predict mutation status without genomic data. I published a paper recently using convolutional neural nets to predict for EGFR mutations in lung cancer histopathology images and compared them to genomic data. But those are examples where we're now starting to use machine learning. Wherever there's a lot of data, you just need a lot of data to train these models. But those are two examples where they're starting to change the landscape, I'll say, over the next decade. Pathology is the biggest, I think, going to have the biggest bang for buck for machine learning.
Maybe another thing how we're using machine learning is and biomarkers is developing prognostic models of disease progression, you know, like having combination of all these biomarkers and clinical features, you know, can that really be used to predict disease progression, which again, is a whole separate topic of its own for an hour talk, but that can be used to enhance trial design, to enhance the interpretation after the fact, for like by adjusting covariate adjustments and things like that. But yeah, so it can be used in multiple ways.
The third example is imaging. In the case of what's your ocular diseases, atopic dermatitis, not atopic. Geographic atrophy, and using machine learning to predict change in lesions in the case of geographic atrophy and provide more quantitative data versus having a radiologist take a look at it and qualitatively see.
So it's based on OCT imaging of the back of the eye. Are there features there that tell us more than the eye can see
Right.
in the eye?
And considering the wide range of biomarkers, what would be an ideal organizational structure or strategy for facilitating successful biomarker/translational studies? I know each company has a different approach to this and would appreciate your perspective.
I think that's a great question.
Probably, I wonder if it's coming from somebody who works at a company. It's a very insightful question. Oh yeah. I mean, I guess that I can start it off just thinking out loud. I think we touched base on the fact that this translational piece is important, like starting preclinically, really understanding the biology, but of the target and the drug, merging that with our clinical understanding of disease progression, and informing trial design, and then like the impact on like the eventual approval. So like you can see how biomarkers has impact in different stages of development with different functions. And so I like to see us as the middle of the universe. Yeah, like we have so many connections in drug development. And so I think regardless of how you organize it, I think the one thing we have learned is we try to avoid as many handoffs as possible. You know, to really not have, you know, one group do this part of development, one group do other part. Like there has to be this continuity. And so you can organize it by disease area, but you know, like, but that's some of the learnings we've done.
Yeah, I'll just add that there needs to be commitment from the company at the executive level to invest in translational science. Without that commitment, the culture in the company does not foster the use of biomarker data to either inform clinical trials or inform research. So I think it starts with executive leadership committing to investing and prioritizing translational medicine in clinical trials. That's number one. Number two, as Heleen mentioned, it is a multidisciplinary effort. And so having cross-functional teams that sit together with, whether it's biostatisticians, whether it's you know, biomarker scientists, clinical drug developers, bioinformaticians, all coming together to say, how do we want to address this? Whether it's from a disease lens or from a molecule lens, but it takes a multidisciplinary team to sit at the table and have equal what's the right word? Have an equal seat at the table. It's not like, you know, you're seen as a afterthought. You know, it has to have an equal seat at the table. And I think that's the only way it's going to work.
Hey, that's all the questions in the Slido. If there's anyone who would like to, oh, there's one. How do you decide which biomarker to use as a measure of clinical outcome?
I guess as a surrogate endpoint or a pharmacological activity. So it comes back to Priti's earlier point. What is your outcome? Are you looking for activity of the drug, or are you looking for clinical efficacy? And so the bar for clinical efficacy is really high. We touched upon that. Like for a biomarker to become a surrogate endpoint, you need a lot of data, you need a lot of prospective trials proving that this biomarker can be used as such. As a proof of activity, pharmacological activity, like the drug is doing what it does, it really comes down again to our confidence, our understanding of the biology of the target and the linkage to the biomarker. If we understand that through a variety of things, the more the better. You know, it would be a good decision to be made, but it's kind of like an organizational decision.
Yeah, I think since you mentioned clinical outcomes, I'll assume it's the former where you're looking for a biomarker associated with clinical activity. It really depends on which stage of clinical trial. If you're looking for a biomarker of clinical activity for a phase 1/2 non-registrational trial, you can use whatever you have confidence in. But if it's from a registrational intent, then it has to be a biomarker the FDA agrees can be used as a surrogate for outcomes.
Well, thank you so much again, Rhea, Heleen, and Priti. Wonderful session, and so great to have you all in the same room. I think you're the only session last year and this year that we're able to actually be live together, and it's so fun to see you talk and really piggyback on all your different thoughts. So thank you again.
Thank
you all.
you.
Thanks for having us.
It was our pleasure.
Yeah, thanks. And
Thank
thank all
you.
for thank you everyone for the questions. We'll see you next time.
Thank you. Bye-bye.
Bye.